Okay. All right. Good morning, Dr. Terry. Can you give me an update on what you've been doing there? Do you hear me? Dr. Terry? Maybe she doesn't. Do you hear me well? Is my microphone working? Okay. Maybe you can start. You can give us your updates on what you've been doing.
Yeah. So EDW is on progress. We have it distributed. And we are working on it right now. And I'm starting last week. I'm helping Dr. Seong with other undergrads on checking up the DICOM files. We're checking the series numbers and we're kind of like matching which image gives like the best resolution. So
Hmm.
we're working on it right now. We're only left with like a few more patients. Um, so it's that. Um, and then on on the update of picture health.
Okay.
Um, so I so we have been uploading like the files for them.
Hmm.
And then we had some calls with um I I forgot his name. But the main contact that we have like with Picture Health, we had some calls and then they told us that the files that is uploaded right now on OneDrive is it's like it's taking like on their end, it's taking a lot of time. So they requested us to um like upload those files on their Amazon Web Service drive.
Okay.
But we don't have access to their account, so we today, actually, right after this meeting, we will contact NUIT and see what we could do. Um,
What do you mean?
but
We can't upload there?
so in order to upload those files, we have to like get an access from NUIT, like to access like their folder, their account. Um, so we have to call the office to get this done.
Why is that?
I'm not too sure. I heard it's regarding those firewall issues.
Firewall.
Yeah,
Hmm.
yeah, yeah. You know, the firewall like prevents us from like getting access to what like Picture Health is using, which is called R clone. It's like their I I I would say program that they are using um to this like to the cloud, I guess.
Okay. Okay.
But yeah, we'll sort this out today with NUIT. Hopefully we could get it done. Worst case, we will just like upload like smaller number of files by compiling all of those images together in one folder so that they could like just download it at once. That's like the worst case. We're hoping to get access of their cloud and then, begin working on it.
Okay.
Yes. Anything else?
Hmm.
I
So
think, yep.
so EDW is done?
No, it's on progress. We're doing it right now. We're working on it.
Okay.
We had like 1,700 patients to screen, so it's gonna take some time,
Okay.
like we're assuming, but yeah.
And
And
who's involved? Other
so yeah, so I'm planning to like include all the undergrads for this screening. Um including Grace, Eileen, and Amy. I'm not sure if Irene with R would be able to do it because like she only comes in Friday like afternoon, and most of the time she's like scheduled with clinic with you on Friday PMs.
Did you get in touch with her?
I did, but I did not hear back from her. Did she not come last Friday?
No.
She did
Eileen, are
not?
you in touch with Irene?
I am in touch, but I'm not aware of her lab presence.
Maybe you can ask her.
Yeah, I'll send her a message right now.
I don't think she responded any of us. So I'm gonna emphasize, if you can't make it to the lab, if you have exam, other duties, if you're sick, let Liam know in advance as much as possible. Okay. I think in my mind, everything is, commitment, keeping promise. If you are not reachable, I think that's a problem. Okay. Anything else, Will?
Yeah, just a quick update on what we talked about yesterday or like the contact info. So Dr. Liam and I are trying to make a spreadsheet for all of those lab members, plus
Yeah,
like incoming lab members to include their phone number, their personal email, and their Katok ID just in case like if we cannot reach out to their phone.
right.
So yes,
Yeah.
we're working on the list.
Yeah. We want everyone to share contact. I've been asking like, oh, can you contact someone? Oh, I don't have phone number. Oh, I don't know how to contact. And then a lot of times Facebook Messenger, I look at it real time, but it looks like it's only few of us. So sometimes it's the phone text or just calling them or even KakaoTalk, maybe a faster way to reach them in case we need to get hold of them. So, all right. So that's that. Anything else?
No, that's it on my end.
Okay. All right, we'll go to Eileen. Any update?
Yeah, we have updates. So Emma couldn't make it today because she's on a flight, but we have a draft that we would like
Anna?
to show you.
Did you say
Excuse me?
Anna?
Oh, Emma?
Emma. Oh,
Yeah. And we have a draft and we have a couple questions regarding potentially replacing Condel, which was the tool that we weren't able to use anymore.
right.
So can we talk about the Condel potential new tools first? So there's four different tools. I don't know if you know them, but one of them is Fathom. I can share my screen too.
Yeah.
You could read this. Oh, okay. Do you see my screen? Okay. So there's four: Fathom, Geno Canyon, MCAP, and Basedell. So this is kind of like what we found from looking through literature, but they're all not like super recent. The latest one being 2018. So I don't know if you have one that you would recommend for us. Also, I know that like another project did like ChatGPT, but I'm not sure. I don't know the details of that project, and I don't know if that would apply for like the things we're trying to do.
I see. So Fathom is similar to, like, it's a go tool like of in itself. Geno Canyon unsupervised machine learning model, which we do not have list. Have to individually run variants will take a long time. Versus Fathom is what's the difference when you run it?
I honestly do not know the details, but I can look further into that.
Ensemble method. We already have a few ensemble models. So right, so MCAP is, just combining different models, so I don't think
Right.
you need that. Bayes-Dell, same thing, so it's either Fathom or Geno Canyon, but not sure if Geno Canyon is machine learning. It could be like ChatGPT.
Geno Canyon.
Yeah. Can you go and
Whole genome functional.
oh, can you minimize your, yeah, I can't read because of your screen.
Oh, do you see it now?
No, maybe minimize
Oh.
your camera screen. Yeah,
Oh,
yeah, that way. Oh, yeah. Okay. Okay. Whole genome functional annotation, unsupervised statistical learning, integrate genomic conservation measures and biochemical annotation data, predict functional potential at each nucleotide. We pre-calculated prediction scores for the entire human genome, and all 22 annotations are available. We have further extended the Geno Canyon framework and developed Geno Skyline, an integrated tissue-specific functional annotation tool. Tissue-specific, Genome Web, Genome-wide association. Which group made this? Can you see where it's coming from?
the Zhao Center.
Zhao Lab?
Yes.
Zhao Lab.
I don't know where they
Where?
are. Oh, Yale.
Yale Biostatistics. Hmm. Can you look at, hold on. Statistical computational method to address, applier method to study biological basis, including lung disease, cancer. We also collaborate with plant genesis evolutionary biologist using NGS and microarray to identify genetic association with various traits. You go down, genome-wide association studies, genome data analysis, including single cell, whole exome, whole genome sequencing analysis, genetic risk network models. Can you go to sequencing analysis? Yeah. Yeah, can you see what they want to do? We develop method to analyze whole exome and whole genome sequencing data, including de novo variants and rare variants to identify genes for both congenital developmental disease as well as complex diseases. This is more variant, protein-protein interaction, mining, what genes are important. Okay, I get the sense what they're. So, how did you find about this one? This
Just like going through literature and just like reading for different. Yeah.
Well, where is the part that say machine learning? Can you maybe go to that? Yeah, maybe software, is that there?
Machine learning.
Yeah, can you actually put like one variant, see how their prediction goes?
Yes. I don't know how to use this. This is a tool that Emma had found.
But that's prediction score.
Yeah.
Where is actually the link to
the tool. I
Yeah,
will find. Oh, no.
score. No, it's a Geno Canyon. Gene Canyon?
Nino Canyon. Mm.
Geno Canyon, yeah. So let's see. Protein variance effect G. Pollution variant. Multivariate.
Sorry, I don't know where I can go to use the tool actually.
Hmm.
Once I figure this out, is it okay if I let you know later today?
Yeah. I think this
Is the
is
best
pretty,
one?
yeah, this looks
Okay.
like one we can use because it's not the same, it's not similar to Align GD. It's
Right.
using different
And
because
Align
it's actually,
GVD had low
uh-huh.
performance throughout all of our tumor types for this
Uh-huh.
project too. So
So I think the the good thing is it's not just the evolutionary sequence conservation, but
mhm,
also it says it incorporated the literature, like annotation,
mhm,
right?
right.
Biochemical annotation data. So it's like ChatGPT, where it's like really incorporating that, whatever's out there, and use that to leverage.
Mhm.
So
Yeah. We'll look
I would
into Geno
say look
Canyon.
into this. If you
Uh-huh.
can't do this, then maybe you can do the ChatGPT. The Dr. Uni and Sally did that, right? So you can get
Oh, okay.
their poster, abstract paper, and kind of
Mhm.
incorporate exactly what they did with ChatGPT. But
I
this
see.
seems to be incorporating
Yeah.
similar approach. So
Mhm. Yeah, we'll definitely look into Geno Canyon first.
yeah.
Okay. Oh, it also said should not be used for commercial purpose without our permission. So I don't know if we have to like contact them to
No,
obtain the
this
tool.
this
Oh,
is
okay.
purely academic purpose.
Oh, okay.
Liam sent something. Check it out.
Oh, I wonder if this is the link too.
Yeah, so if you go into this link, you can there's like a
Oh,
download button. You can
oh,
actually
awesome. Thank you
download
so
the software.
much. Yeah.
Yeah, maybe
Yeah, I'll
download
figure
it
this
and
out. Mm-hmm.
you can practice.
Mm-hmm.
But is it for each chromosome? I was just looking at. Anyway, learn how to use it, maybe,
Yes.
um, and see if it's something you can learn.
Mm-hmm.
Yeah.
Will do. Thank you.
Bye.
And then draft. I think you did review parts of it before, but I know the results have been updated and the discussion.
Mm-hmm.
If you want to see that first, or if you want to just read through the whole thing.
So just show me the ones you corrected, or
Okay. A lot of it was just replacing our values
okay,
because the results were pretty much the same.
okay.
So this is like our new table, and then we have more tables when we go to this link.
Okay.
I'll go back to the draft.
So you updated the numbers,
Mm-hmm.
and then you have colorectal and melanoma here.
These are potentially going to be removed because we still got undefined MCC scores for this, even though we like lowered the exclusion criteria.
Hmm.
So that's also something we can discuss.
Why did it get undefined? Because
on,
we're lacking something?
that's probably the case, but I'm not sure exactly. But colorectal, sorry. We got 0 for MCC and then undefined for MPV. And then melanoma, we also got undefined and 0.
Hmm.
Mm-hmm.
And go up on the top of the table.
Yes.
So accuracy, MCC, specificity, photo variants. So why is Conda? Oh, Conda we
Oh,
tried
yeah,
to
that's
take
the
out.
one. Yeah. So
And
that
then
would either be
yeah, for total variance,
yes.
what's the denominator and
1,161
so
variants.
that's total
The
number of
total.
variants?
Yes, from all the different cancer types.
And what's the different numbers?
So like breast had 823, ovarian had like 706. Is that what you mean?
No, the 140, 159.
Oh, because
What?
they weren't all able to be used. If that
Mm,
makes sense, like they wouldn't run through the tools. So these are the ones that had to be. So for Align GBD, for example, 21 out of the 1,161 were not able to be run.
mm. Did you explain that in the method?
Not explicitly, I think, but we talked about like our exclusion criteria kind of, but we didn't. I think that's something to put in our discussion, or is it in our methods?
I think you should put it in both
Okay.
because people would think, why wasn't it able to be run, all that.
Mhm.
And going back to the table, go back to the table. So here, so overall performance, it may be better to put like for overall performance,
Mhm.
you can, and next to it, you can put n equals 1161. And then for Align GVHD, you can just put the number 1140,
I
CAD
see.
1159, because
Uh-huh.
having the same denominator is confusing.
Did you mean n equals 1161 here, like at the top, or do you mean like overall performance, like here?
There, because
There.
it's
Okay.
gonna be different for breast cancer.
Right. And then each cancer type also had like variants that we cannot do. So
Yeah, so same thing, breast cancer,
uh-huh.
next to it, you put n equals 823.
Got it. Thank you.
Yeah. It's confusing to see
Mm-hmm.
what that really means. And maybe at the bottom of the table, you can put in the note,
Mm-hmm.
yeah, why like total numbers were, you can put that explanation why those numbers are all different by by the algorithm, and yeah. Okay. Okay,
Okay.
continue on.
Mm-hmm. Bring back to the results.
I think you can still put colorectal melanoma, but not just
Describe.
don't mention the ones that you don't have the output. but just mention the ones you have them output is fine.
Okay.
And again, what journal are we aiming for?
Oh, I don't have the list on me. Emma has that.
Okay.
Yep. I can message that to you as soon as I reach her.
Yeah. It looks like eLife is another one that kind of broadly accepts papers like this.
Mhm.
eLife as or scientific reports are the ones that can be considered.
Mm-hmm. And I know you also discussed like there's like temporary journals that you like submit to while like it's waiting to be published, I think.
Yes,
Last
so
week.
that is called BioRxV or MedRxV.
Okay.
So I would submit once you're done with the paper,
Mm-hmm.
submit to those are called preprint, and it will be
Okay.
published online. I think it's important to do that these days so that
Mm-hmm.
it's
It's
there.
still out there, right?
And then you guys can all put that in your resume
Mm,
as like a preprint publication.
right.
It's better than put, manuscript in prep.
Mm-hmm. Will do. Thank you.
Okay. Okay. All right. The discussion.
Mhm.
Okay, without. Okay. Limitation. Yeah, it's okay.
Mhm.
Yeah, I like it.
Thank you so much. So once we figure out how to use Geno Canyon and whether that will be implemented, we'll add in the data and then final
Mhm.
confirmation. Mhm.
Mhm. Good.
Thank you.
Yeah. So I like how you wrote this in a more, I guess, scientifically sound way.
Oh, thank you.
Scientific writing is difficult. Some
Yeah.
yeah, it can be sound too colloquial or too extreme, promising we have to do this, or it's like more caution and euphemize
Mhm.
as much as possible. So that can be done by reading many different papers. Like, oh, how do
Mhm.
they, what words did they
Right.
use? So medical writing is pure repetition exercise, and you're gonna get better the more you write it. So
For
good,
sure.
good job.
Thank you for your help.
No problem. So Eileen,
Yes.
so in 3 weeks,
Yes.
well, it's in 2 weeks maybe. There's an AACR
Mhm. Right.
and it's in San Diego.
Uh-huh. Is it next year, right? In like springtime?
Yeah, April.
Mm-hmm.
So who is your team?
For this project,
Yeah, Eileen, Emma.
this is, yeah, me and Emma, and Amy also helped with the results part recently.
Okay. And who's the corresponding author for this?
I can show you, but uh we haven't updated it since when we like first started it, but right now Dr. Chu, Dr. Eugene, Emma, me, Grace Lee, Alex Lee, and Dr. Che are currently listed.
Okay. So ones who did not, well, I guess whatever if they have contributed, in a way they deserve to be author. Well, right now you're the project lead, right?
Me and Emma.
You and Emma, yeah. You let me know if if they still deserve to be author or not, depending on
Right.
final paper, like their contributions, and what data they collected and all that. So
Yeah. I'll talk with Emma about that too.
yeah.
First.
And whether you do ChatGPT or the Geno Canyon,
Mm-hmm.
maybe just the data you collect
Mm-hmm.
and just that concordance that you get out of it, which is going to be updated with other algorithms.
Mm-hmm.
Maybe that updated portion you can write it as an abstract.
Just on Geno Canyon.
Yeah.
Or okay. Yeah,
Yeah.
I'll look into
Either
that.
one. If Geno Canyon works, then great.
Okay. And if it doesn't work,
Then ChatGPT, do ChatGPT because that's the machine learning.
okay.
And write that as an abstract and submit to AACR. I think it's still 2 weeks left. So divide and conquer.
Okay. That sounds good. Thank you so much.
No problem. Okay. Dr. Seong? Oh, before going there, Dr. Seri, are you able to give us update on what you've been doing?
Yeah, sure.
I can't hear you.
Can you hear me now?
잘 안 들립니다, 아직도.
Um,
나 한국말로 하셔도 돼요, 또. 지금. 근데 안 들리네. 마이크가 잘 안 되나?
can you hear me now?
아직도 안 들려요. 아니면 전화로 하셔야 되나? 마이크로폰을 바꿔보시고 안 되면 다른 기기로. 이거 에슬리도 선생님이세요? 에슬리는 누구죠? 음. 안 돼요? 안 들려요. 안 들려요.
It's not working now.
어, not working은 조금 들렸는데 잘 뭐가 안 되나 보네요.
I was just
전화기로 보통 하면 되는데 전화기 안에 있는 앱으로 해도 되는데 컴퓨터가 안 되면. 네, 일단은 해보시고요. 고쳐지면 다시 돌아오겠습니다. 네, 이세영 선생님.
네. Um, I'm trying my best to share what I've been going through neatly, but I really did my best, but I'm still kind of in a mess. So I hope what I'm conveying is clear enough.
음.
So 3 big things I would have gone through this weekly. First thing, very light. I helped the logistics for Dr. Chian, the ZC 2023 Radiomics poster. I did some modification based on Dr. Chian and uploaded on 25th of October. And Dr. Liam helped me a lot in this. Second thing is the immunotherapy analysis based update. I wasn't able to update. I was able to update some, but I couldn't organize it and come here with a slide. Basically, what I did was after checking on with Dr. Che last Friday or so, I did the RECIST and iRECIST and durable response and mixed AB analysis for the intrathoracic, which was 197. So I matched up the systemic 205 to 197. And the outline was that from these patients, for evaluating mixed response, I realized that these patients should be also categorized again, whether the patient has single lesion or multi-lesion. And from this, I recognize the smallest group to, if we were to compare that as one N number, the multi-lesion patients for the intrathoracic patients would be 142. And
That makes sense.
yeah, so yeah, I would so these mixed response rates were the things I showed you Friday.
Can you go back?
Oh, this.
So systemic lesion, single lesion is 24.
Yeah.
Intrathoracic, single lesion is 45.
But I think I have to check on these numbers again because I just noticed that these don't match up to be 97.
Yeah, how come 45 is bigger than 24 up there?
Yeah, I'll have to check.
It has to be smaller than 24. If you think,
Right.
because intrathoracic is, you know, smaller than systemic, so
Systemic.
you understand, right?
Intrathoracic is smaller than systemic. So do you think it should be smaller? I think it should be bigger.
I'm saying single lesion in systemic is 24.
Yeah.
Single lesion in
Right.
thoracic should be smaller than 24.
When I was organizing this, I realized that intrathoracic single lesion patients increased because, for instance, if a patient had one intrathoracic lesion and then 2 other lesions were from maybe liver, then the liver lesion would be gone. So the patient would only have 1 lesion, which would be only an intrathoracic lesion.
So, so this is the difference in how we perceive so so let's think about this. Systemic lesion. There's a systemic lesion, but there's zero intrathoracic lesion.
Yeah.
For those patients, what are we using to predict? Nothing, right?
Yeah.
We're segmenting and we're using only intrathoracic lesion to predict. If
Mhm.
there's no intrathoracic lesion in this A patient,
Mhm.
we have no input, correct?
Yes.
We're not segmenting anything.
Yeah.
So when we do analysis, did we look at 205 or 197?
You mean the analysis using radiomics? I think it's only 197, and this decreases even more because, from my understanding, radiomics doesn't use lymph node lesions, and there are patients who only have lymph node lesions.
So when you say intrathoracic, this includes lymph node.
Yeah,
Okay.
because I was doing this analysis just for iRECIST, RECIST, and mixed and durable response.
Okay. What is the title of this project again? It depends on what we're interested in.
This was actually something I got from Dr. Tegu, and from
it's
what
Mixed
I
response.
think, yeah, I think it was just the clinical responses.
Yeah, so there's no radio involved here. It's purely clinical, right? Then it makes sense to, well, we're asking the question. So intrathoracic is going to be a subgroup analysis. The mixed response phenomena, is it still relevant, or is it more relevant or less relevant if we look at only intrathoracic lesion? So that's the kind of question we're delving into. But the
Yeah.
systemic lesion is the wanted analysis that we want to do. So,
Okay.
and it might be more true if you have one lung lesion, one liver lesion, you are able to analyze whether there is going to be mixed response. If you only have one measurable lesion,
Right.
you will not get mixed response.
Yeah.
You will not. So there is no point asking this question if you have single lesion, right?
Yes.
So, assuming all these numbers are true, then 181 is the magic number we want to use. And
Okay.
after we do all these analysis, then we can say, what if we limit patients with only intrathoracic lesion? So I want to say intrathoracic lesion, is that and or you know what I mean? Is it at least one intrathoracic, or everyone has only intrathoracic lesion? When you say intrathoracic lesion patients.
Oh, it means only intrathoracic lesion. So every extra thoracic lesion was deleted.
And that's only a patient?
Oh, so 8 patients only had extra thoracic lesions, and 197 had at least one intrathoracic lesions, but from those patients, extra thoracic lesions were cut out for RECIST or iRECIST response.
I see. So I think you have to make that clear. At least one intrathoracic lesion patients were 197. And of them, when you say single lesion patient,
Yes.
Do you mean single lesion only in intrathoracic or anywhere out of that 197?
I'm sorry.
So 45 is single lesion where? At least one thoracic lesion? Meaning what?
Oh, so these 45 patients would only have one lesion that is intrathoracic.
One lesion only in intrathoracic, okay. So I think we have to make that clear because I'm not out of 197. If you say only, if you choose only one lesion patient, that's not conveying the right message. So, single lesion present only intrathoracically, including lymph node and intraparenchymal. So you have to write that out in the method, what those patients are.
Yeah. So basically, I think I just want to make this clear. So for example, if a patient A had one lung lesion and two hepatic lesion, that person would become a single lesion patient here because the hepatic lesions are removed for the analysis for this.
No, it's not removed.
Oh, it's not.
It's
Removed.
not.
Then what are we?
So I
Then
think, so I think we have to revisit the question. What question are we asking?
okay.
Oh, it's the first snow of this year. Or this
No.
yeah, it's snowing outside.
Wow.
Eileen, you should take a look at the window.
Yeah, Eileen, you should come over. It's amazing.
Yeah, you can't see it from where you are.
Wow.
Yeah, it's a lot of snow.
Ugh.
For
Lug-
snow. It always snows on Halloween.
Yeah.
Always snows on Halloween.
Wow.
Oh.
Everyone's pumped.
Yeah, it's snowing. It said it's gonna snow.
Right. So I just want
Maybe go
to,
out.
I want to
Make some.
snowman or
That one
something.
too.
Is it going to snow whole day?
No, just for an hour or so.
Will says just for an hour or so.
Okay, we should take a break in 5 minutes so that we can go out.
Okay.
Anyway, so let's finish this part. So my question is, if you have lesion in different organs,
Yeah.
they are so different that even if they receive same treatment, they might respond differently, shrink or still grow. And if you have that as your predictor, independent variable, that might be able to predict how long patients are going to live. Now, if you want to only look at what's going to happen inside the lung, are you going to be able to predict this phenomenon better or? So that's the question. If you want to ask that question, you have to limit to a patient who only has lung lesion. If you have liver lesion and exclude that liver lesion, you're kind of ignoring the biology, what's happening there. So in you have to select patient with all lung lesions, not at least. I'm talking about patient with only lung lesions, nothing outside lung. And then we want at least 2 measurable lesions, whether it's lymph node or lung parenchyma. And ask that question. It's the same question. So your subgroup is going to be very small. No distant Mets outside thorax as a subgroup. And then if you confine to that subgroup, are you going to be able to say mixed response at the first scan correlates better or worse with overall survival? Do you understand?
I do understand that the subgroup should be patients with only interthoracic patients and with 2 or more lesions to analyze their mixed response. But now I'm kind of confused why, what are we looking after using the rest? We're not comparing those two.
What do you mean rest?
So we started with 205.
So that's what we want to do. Well, 181 is we want at least 2 lesion because 24, these are people who will never get mixed response, right?
Right.
So we don't want them in our analysis, to be fair.
Okay.
So this is what we want to do. 181 is the main analysis. Now, if we want to have a separate question, what if we only look at patient with thoracic lesions? Will we see this pattern in a more clear way or less clear way? Do you
Got
understand?
it. Yeah, I understand.
Because lung, if it hasn't spread, so this to me, do you have many distant Mets? That means your tumor is more heterogeneous. It requires a lot of more mutations to be able to be equipped with metastatic potential and grow in liver and bone. You need a special gene mutation.
Right. Okay, I kind of get it. Because when I first got this data, I was in the meeting when Dr. Taegyu was going through all these lesions to check with Dr. Jiyeon whether they were interthoracic or extrathoracic. So yeah, so from that point, I was guided that like I would want to remove these lesions independently and then run another analysis. But I guess it's just
Yeah. So
getting down to this.
what makes biologically sense? What makes
Yeah.
number 2, that's why the team has to be in constant communication with me. Okay. I can say let's do this and you might get it in the wrong way. Do not guess. If you are not clear, come and ask. And even if I say it, sometimes it's clear to them in a different way.
Okay, right.
Constant communication is key. And I understand Dr. Jiyeon, Dr. Taegyu, they're in Korea, they're very busy. That's why we need point of contact to make sure it's in line with our theme.
Okay. So to wrap up with this part, for the future plan for me was to do a mixed response per iRECIST or RECIST subgroups. But for now, I guess the group would be mainly this one, the first one, and then secondly, this cohort. Am I
Right.
correct? Okay. So I'll be doing mixed response analysis, durable analysis, and then MPFS, MOS, and hazard ratio for the 2 groups, the main and the sub.
Yes.
Okay. And the main would
181.
Okay, so there would be no comparison between systemic and interthoracic.
What do you mean?
Because before I thought we were kind of comparing the systematic results and interthoracic results, but I guess we're using the same lesion, so there's no way the results would be different.
What do you mean they're saying we're doing intrathoracic lesions only group?
Right, but like the intrathoracic only group would have different number of patients than 188, so 181. So we can't compare those two, right? It's just for better understanding, but not one-to-one comparison.
If you have a different subgroup, you're not going to have one-to-one comparison, but you will be able to see what's different in all these parameters.
Right,
So you can
Right,
stick to the numbers here.
right. But like, I was just confused because last time when I received, last time I updated you that the intrathoracic patients are 197. So the systemic would also have to match 197. So I also thought those two analysis would be different, but for the understanding now, if the number of patients are the same and we're not excluding the extrathoracic lesions for the intrathoracic analysis, there's no point of like this. Just I would say I can fill in this, but like the interthoracic number would be just very, very small here and 181 here.
Right.
Okay. Yeah.
Yeah,
got
but
it.
still fill in the number. I do want to compare.
Sure. And one more question. So I was wondering, we are using the same patient group as the radiomics, and I thought this was kind of related. So from what I know, the radiomics prediction analysis was done on the prior data of RECIST and iRECIST.
Mhm.
But this clinical analysis has been very much updated. So for further radiomics analysis for the response prediction, are we looking forward to updating these results on those analysis, or is it just being a very separate project?
So I think it's related. It is related. Only as so radiomics, when it's looking at mixed response,
Oh.
So radiomics is asking different questions. So this is like you are using baseline scan and the first scan as an independent variable. We have our own definition of mixed response, and mixed response is what's going to predict overall survival. Okay. In radiomics, your independent variable is only baseline scan. And you want to predict if the first scan is going to show mixed response or not. That's your outcome. Okay. That's your outcome right there. So you're asking different questions. But you will never get mixed response if you have measurable lesion as a single lesion. So I think, so for radiomics, just that's predicting response alone, it's okay to use one lesion, single lesion. But if you are at endpoint as a mixed response, I think it makes sense to limit to only 2 or more lesion patients. So in a way, that's similar. And then in that mixed response, are you going to be able to predict mixed response better by using only the baseline radiomic features? To answer that question, I think you need the same intrathoracic lesion only group.
Right.
So only asking that question, you can use the same clinical subgroups here. Do you understand?
I think I followed most part, but I'm kind of confused on the last. So, what I was thinking was that the overall survival result should be the same for the clinical analysis and the radiomics, right? Because we're using the same patients.
So radiomics model doesn't predict, at least now, not now. You can write your own algorithm based on the shared AI source, but it's hard to predict PFS or OS.
Oh, okay, so we're just
Model
focusing
is
on the
1 or 0. 1 or 0. Are you gonna have durable response or no? Are you gonna have mixed response or no? So the output is 1 or 0
got
for
it.
all radiomics related analysis.
Okay,
There's not gonna be hazard ratios.
okay.
Now, what we are able to do is if the radiomics gives like durable responder as 1 and non-responder as 0, we can
Mhm.
use that to separate PFS curve and OS curve. And that's what we've been doing. Do you understand? And
Yours.
that's very different.
I do understand that the algorithm can only predict 1 and 0, but I didn't get the durable response 1 and 0 and PFS and OS. Are we doing that too? Because I haven't done that yet.
We have to do that. Part of the paper.
Oh, so just like mixed response is 1 and 0, we would want to do analysis of MPFS and OS based on durable response too.
Right.
Oh, okay, got it.
But this Tegus paper is only looking at mixed response.
Okay.
So we're doing that, if you have mixed response, SD group, PR group, PD group, what is PFS, what is OS? It's a clinical paper. There's no radiomics here. But when we go to radiomics, if radiomics predicts mixed response, would that affect OS and PFS? So that's a separate question we're asking in radiomics paper. If the radiomics predicts durable response, 1 or 0, does that predict change in PFS and OS? That's the different question.
Got it. So the clinical analysis would be that does mixed response or durable response affect MPFS or MOS? And radiomics would be predicting whether durable response or mixed response would be made.
Right. So durable response is not part of this paper. People are not interested in the question because we already know. We already know that RECIST criteria predicts PFS and OS. That's why even RECIST criteria matters.
But we're doing the analysis just for a stronger paper? Why?
No, we are so going back to this very specific project. Having mixed response, does that even matter? What does that mean? That's the question. What is the meaning of having mixed response at the first scan? We don't care about radiomics. We don't care about RECIST. We don't care about durable response here. We care about mixed response. What does that mean? Does that mean patients gonna die sooner? Does that mean your therapy is going to not working sooner? There's not many paper asking this question.
Right.
Especially with the immunotherapy, especially with lung cancer alone. No, you have to keep reading about similar papers, what other people have done,
Okay.
and see why is our question unique. That makes sense?
Yeah.
Okay. So when you're not sure, go back to the question. Why are we even asking this question? Is it worth my time and effort?
Right.
Yeah.
So for now, do you still want the durable response analysis as in 0 and 1, or is it just an extra for now? Like I do have these are just not I can calculate this without much effort, but to analyze this MPFS and MOS, it would take some time. So
So that was part of the radiomics project.
the durable response.
Yeah, my sense is that durable response has already been calculated from the radiomics project. And again, that's not part of this project,
Okay, I get it.
but you will need the resist category because we want to look at that within the context of each resist category. Because the mixed response itself isn't any, it doesn't have meaning.
Right. Okay. I have like quite a question, but the snow is
I know, I think.
disappearing.
So we'll take a break. Let's take a bathroom break.
Yeah.
Back
Sorry.
in 5-10 minutes.
Okay.
Liam, are you there?
네, 돌아왔습니다.
Dr. Che, can you hear me now?
Oh yeah,
Ah,
hear you well.
so sorry. It was very embarrassing.
Oh, that's okay.
Yeah.
I don't know if you guys see it, but it's now snowing a lot again. Can you guys see the snow?
I can. Eileen and Will should come here.
It is?
It's snowing a lot. It's the first snow.
Oh, I see it. I see it.
Yeah.
Oh my god.
오,
too.
많이 오네요.
Wow.
아,
Yeah. All right.
눈
This
엄청
is our
많아.
present for people in Korea and in California. Wow,
Thank you.
it's a big, thick snow now. Um, and it's Halloween today.
Oh? Oh,
It
I
snows.
didn't realize.
It snows in
Wow.
Halloween. Okay, we're back. Well, we have people from Korea, Chuhee and Dr. Isers here.
Mhm.
So maybe before it gets too late, let's do them first. Chuhee, do you have an update?
I don't have much updates, so I didn't hear back from Lancet regarding preprint, so I think we're just gonna go ahead with SSRN.
What is SSRN?
Apparently it's a huge preprint database. They deal with all sorts of I don't want to say departments, but different things, not just medicine. So they have social sciences like economics. They just have everything. It's a really huge database. But I'm
Show us,
I'm
share your
sorry.
screen. You show us.
Okay, one sec. Let me just. Do you see my screen?
Yeah.
So you can just look at like different they have accounting or agricultural science and everything. And I went ahead to look at their so they have a special section for medical.
Yeah.
Which is what I think we should submit to for preprints. So they seem to be a huge database and there seems to be a lot of people submitting.
Do they
I
accept case
Huh?
report? Do they accept case report or me?
I think they do because they when I was going through the submission process, I haven't finished submitting it yet, but when I was going through the process, I had to tick mark for case reports. So I think they do accept case reports too. I don't know how their review system is, so I don't know if they have an intensive system where they have all these standards or if they let everybody submit and preprint. I'm not sure about that. I was just gonna wait for Lancet to get back to me, like give them a full week before submitting to SSRN.
Okay.
But if Lancet doesn't get back within a couple of days, I was just gonna, with your approval, and I was gonna ask Eugene and Isel and see if they're okay, just go ahead with SSRN.
Okay.
It seems pretty, if you look at their statistics, it seems pretty like a lot of people contribute.
And who owns this site?
One sec. Elsevier.
Oh, okay. They're publisher.
Yes. It's funny because I think I had a preprint with them once because they remember my information. So yes.
So
Okay.
I think they are a legit site. I think it's stable. It's renowned. So if Lancet doesn't get back, then I think SSRN is a good bet since the Med or XIV don't take in case reports.
Okay. That is fine. Let's do that then.
Okay, sounds good. I'll pass it on to Iso then.
Yeah. So let's put this as our new SOP for publication. I wanna now
Oh, okay.
yeah, for any publication, including case report, original wow, it's a lot of snow now.
Oh, is it?
Let me show you guys right here. It's a lot of snow. I don't know if you can see it.
Oh, wow. I do see it.
It's a lot of snow. Wow. Anyway, I was gonna say same thing with Eileen, your paper, and Isol, your paper, Liam, but not anything that comes out from our lab, not like SWOG or we will be posting. So my preference is if we are submitting to Lancet related journal anyway, we'll use Lancet preprint. That's number one. If not, we're gonna use medRxiv. And if they don't take it for whatever reason, then we'll go to SSRN. And medRxiv is any human data related. If there is just biochemical data, they have bioRxiv. So they're equivalent, they're same thing. But I guess Eileen, Covariance project, they're all coming from human cancer data. So they're more medRxiv. If they don't take it for whatever reason, it's SSRN, okay? So for anyone who hasn't published their paper yet, let's make sure you have it published in the preprint world so that at least we can claim that we have made our publication available to the world at certain given time, and everyone can look it up. And more importantly, you guys can put that in your CV and say it is published in online as a preprint. But it's also submitted to an academic journal at the same time. I think that will carry more weight, and people can actually look it up by just going into that website, and you can put that website address in your CV, and that's going to make it more presentable. So, yeah, so Liam, I want you to make that SOP available to everyone and make that a shared Google Doc drive. And Will can help Liam, and then share it to our Che Lab messenger chat room so that everyone knows our new policy. Okay. And then yeah, definitely also share the new contact information Google Doc among our lab meeting, so that people can fill it out and you guys can follow that up. So okay, that being said,
Sorry, I can I add something? Sorry.
go ahead.
If you're done. So for the cancer screening, it's still under peer review, and I think we should wait a little longer.
Cancer screening.
At Scientific Reports, it's under peer review.
So
So yeah, that's it.
okay. Okay. And you're gonna meet with Dr. Iser to talk
Yes.
about new projects?
Yes.
Okay. Okay, thank you. Iser, are you available for update?
I am. So I just want to discuss these patients under with the PDL1 level below 50 and then who got the immunotherapy only regimen.
Mm.
So I looked up each and every patient. So among those 44 patients in total, so they have like PD-L1 level low or negative, and then they were treated with IO only regimen. So 20 patients got ipi-nivo, which is a part of the standard frontline, and then 14 patients got pembro only. So the rationale there I found was where was it?
So these are PD-L1 negative
Negative or low.
or low. Okay.
Yes. Keynote 042 press release has just shown that there is OS benefit to single agent pembro as frontline for PD-L1 over 1%. So this was written in the like document as like a rationale of choosing the pembro for some patients having PD-L1 like 10%, 20%, and then other patients like there were like documented reason mostly like patient did not want chemo.
PD-L1 negative, but limited sample. Oh,
Yeah. And then patient did not want chemo. This patient given PD-L1 positive, so pembro only. Then this patient was opposed to chemo as well. This patient had like myelosuppression.
wait. Patient received dabrafenib trametinib before
Yeah.
and then got pembro as a second line.
Yeah.
But this is sort of like a first I guess the first systemic. So, well, I guess it's okay to use it as a, still call it first line, right? Because is this first line only paper?
No. We're actually we're actually including every lines of treatment.
Oh, okay.
We did do analysis on the subgroup analysis of frontline, like first line versus second line and beyond and all that. So we're adding the criteria that we're choosing the regimen of interest is like whichever immunotherapy that is closest from the blood draw for the Veristrat.
Okay. So do you think Veristrat was drawn before dabrafenib or after dabrafenib?
So the sample draw date was 2016 November, and then treatment initiation was, so it was after.
After dabrafenib?
Yeah. So after dabrafenib, patient progressed, and then we started the treatment, and almost right after starting the treatment, we drew the blood.
I see. So this patient was pembro alone and PD-L1 low, or
PD-L1 was 1 to 50%. We did not write the exact number. Maybe we did.
0%, maybe 0.
Let me just double check that. It's like annotated incorrectly, I think, because it's annotated as low. This has to be like 1 to 50%, and then this numeric value is
0.
0. I'm not sure. I can double check that.
Okay.
But for every patient like who got the pembro only regimen, like there was this written reason, like
Let's go one by one.
Yeah. So this patient got BRAF
Yeah.
mutation and then progress and then pembro. And then this patient got carbopen pembro and then got the myelosuppression from the cytotoxic chemo. So as a second line, we're starting the pembro monotherapy.
So this is Carbopen Pembro followed by Pembro is one. It's not immunotherapy only.
Yeah, so what we're doing is IO only regimen, when the regimen of interest, which is the closest ICI maintaining ICI containing regimen that is closest from the blood draw. So this patient is getting IO only therapy as like a regimen of interest. That was what we did.
So do you think patient got one cycle of Carbopem or something like that?
No, so this patient got Carbopen Pembro previously, and then had this did not tolerate it well, and then had to change the regimen, and the sample blood draw was November 2018, and then the index treatment was April 2019. But we chose this Pembro only treatment as a index regimen because these Carbopen Pembro was started far back from the blood draw. So this Pembro only regimen was closer to the blood draw. Like that's how we just set the rule. Like there are patients who had these multiple lines of treatment. And then some treatment is like far ahead or after the blood draw. And then this is not the like prospectively collected cohort. So we had to just set the rule to pick the regimen of interest. And then that's how we just did like the closest ICI containing regimen from the blood draw. So it's like not necessarily like ICI naive or anything like that.
I see. How far do you think it is from like Carbopem being used?
That I did not check, but I can take a look. But like if that's like if that becomes our concern, then we'll have to check all these 173 patients because like there are patients who had like multiple lines of immunotherapy, and then for all of them we chose this regimen of interest like this way.
Oh, I see. So let me tell you my viewpoint. If someone starts with Carbopem Pembro and continue Pembro onward, and then Carbopem had to be stopped because of infection, myelosuppression, that's still part of the combination regimen. Because sometimes we stop carbo only, right? Sometimes we continue pemetrexed, but sometimes we develop kidney failure. If they really bad anemia, we stop pemetrexed. That's going to happen, but it's still and then patient might end up being Pembro alone. But from the start of the VERY-3 regimen till the end of the monotherapy Pembro, it's still considered one regimen.
Yeah, you're right. I think that makes sense.
What happens, even though infection happen, whatever happens, that's how, if you think about all the keynote studies, that's how they still measured combo results.
Yeah, I think I got your point.
Yeah, so I feel like yes, it's a monotherapy that started, but I feel like this patient should belong to still combo. If they got at least one cycle of carbo platinum doublet, go to combo regimen. I hope there aren't that many.
No, like for all the patients who were classified as IO only, I looked each and every one. So for that issue, like we can just go through everyone. So for this patient, the biopsy PD-L1 was negative, and then they ordered another Tempest PD-L1, and it was 98%.
Wow.
So patient was started on Pembro only. So this one I was kind of confused because looks like when we were like collecting the data, when we just looks like we just checked the path report and then like not the documented PDL1 in the like additional documentation. So I think that's why this patient was classified. Uh-huh.
I'm interested to know if they looked at exact same tumor or they looked at different biopsy. Maybe.
Yeah, so that's actually what I was trying to find. I actually looked up the Tempus report itself. It didn't show the biopsy site there.
Oh, really?
Yeah.
Hmm. Sometimes in the path report from our own path department, we'll say the sample sent to Tempus, or something. But
Ah.
yeah, that's how you know. Yeah,
Yeah, but for this, go ahead.
but the Tempus, even if they don't say it came from liver, lymph node, the bone, like a lot of times it has sample ID. But I mean, I just want you to do a deeper search. Why is there a difference? Because we know, we know from learn from the case. And just let me know on your later on next that meeting or whenever you find it, just send me a messenger message. But for the sake of this research, so we don't, so for all of you here, PDL1 matters within the within the tissue structure, stroma, and then the tumor, all that's important. But if it's FNA, or if it's a cytology from pleural fluid, it becomes artificial. It's gonna be very difficult to have a accurate presentation of PDL1 expression. So it matters if our own in-house PDL1 and Tempus, if they looked at different tissue and one is FNA, one is more of a biopsy, one is cytology. Cytology, I don't count that. I mean, it means something positive or negative, but it doesn't tell me whether it's low or high. So maybe it's better. It's good to prudent to look at the source and find out why is there such a big difference. Like maybe the biopsy happened in more necrotic tissue, and maybe it's really just not a good representation. Because if you look at the in-house PDL1, like if you look at the tissue and says extensive necrosis present or something like that, then it's not accurate, right? So maybe do a deeper dive. Which tissue is better? If it's coming from exact same tissue, I think there's a discrepancy. And patient got, and then maybe after reviewing that, we can go with whatever sample that has the best quality to assign this patient, whether it's gonna be low group or high group. Let's revisit this patient next. Okay. Okay.
And this patient as well, the in-house PDL1 was 5 to 10%, and Tempus PDL1 was 50%. It was started on Pembro.
I see.
Do you want to go through this as well?
Yeah. So for this patient and the one before, let's say if you do all the search and you don't see meaningful difference in terms of sample quality, because the decision was made based on Tempus, and I do think Tempus, I do believe they have pretty good quality. We will go with the Tempus because we, and then we assign this patient to high group.
Gotcha.
Okay.
And then this patient given PDL1 20%, and given patient is So he basically did not want to receive the immunotherapy, and the
Okay.
PDL was 20%, and then was getting this radiation treatment. So he was just started on the pembro.
Oh. Is this stage 3 patient or stage 4? Do we know?
Stage 4.
Stage 4. Do we know if there's a distant mets or we don't know?
No, I don't think we collected that much data.
That's stage 4? Okay. So I think this is a good data. So like among PDL1 low, high, we can kind of among this patient, what percentage refused chemo or something like that. That of in itself is a worthwhile data to report.
Sure. So when we describe like these groups, like we can just briefly mention like how many patients refused the chemotherapy and then what was the rationale for the pembro only and all that.
Yeah.
So yeah, basically, so this patient, there was no like obvious mentioning about like what patient feels about the chemo or anything. It was just given positive PDL1. These patients, there was
That's based on so like just like the keynote 042. So for you guys, 042 is the well, there's 042 and 421. Just okay. So keynote 042 is the ultimate phase 3 randomized data. That led to approval of pembrolizumab. Show you. All right, can I share?
Mhm.
Okay. Oh, where is name? Okay. Do you see it?
I do.
Okay, I'll make it larger. So keynote 024, Julie Bramer at Hopkins, 2020, mixed histology, mixed meaning squamous and adeno. You have pembro compared to platinum doublet, 1-to-1 randomization, smaller sample size. Oh no, how do I move this? Wow, I can't. So hard to control this. Where did that table disappear? I don't know what happened. That very table I was looking at, I can't find it anymore. Oh, here. So Julie Bramer, keynote 042 is it's a Pembro, same thing, but bigger size sample. And they use the 22C3, so median follow-up. So this one, Tony Mock from Hong Kong University, Chinese Hong Kong University, keynote 042, bigger randomized. This one is the one that showed 7. That 1%, if you do the 1%, it does make difference. It didn't really do a good job here showing. I want to show you a subgroup analysis here. Let's go post. So this was an ASCO plenary paper. And if you do subgroup analysis, the effect was greatest among patients with PD-L1 50%. So this is 1,200 patient. So you can look here. Pembro chemo overall survival median was 20 versus 12 with TPS high, and it was 17 versus 13, TPS 20. 1% was 16 versus 21, still with good p-value.
But my
So-
question is like, isn't our question like, is Pembro only like not inferior to Pembro plus like chemo?
So if you look at the low PD-L1,
Mhm.
13 versus 21 with, look at the p-value is not significant, right?
Yeah.
So it's not, well, I guess non-inferior is okay for this.
So my question is like, didn't they just compare Pembro versus chemo? But like, are we like questioning is Pembro versus Pembro and chemo like combination?
That was asked in a different trial. So this trial is only Pembro versus chemo. Keynote 1 and 42 only ask the question Pembro versus chemo only, and the answer is in 1 to 49%. There is no superiority. There is non-inferiority, but there is no superiority. So that's why Ian says if it's low, we don't recommend using Pembro alone.
Yeah,
That's
exactly.
what Ian is saying. But what your comment in the Excel spreadsheet box is FDA approved it, based on this number, combining low and high, it is still significant, right? So it is FDA approved, but it's not NCCN
Yeah,
endorsed.
it was not reflected in CCM. I just copy paste that sentence from the fellow's note. So that was like it just sounded like that was the reason that they decided to give the Pembro only to that patient, even though the PDL1 was like not over 50%.
So I think that's so you can use so for those, I think you can put it as the at treating physician's discretion.
Okay.
Because when the physician believes that well, it's going to be at least as good as chemo. You know what I mean? It's not inferior, right?
Mhm. Gotcha.
So I don't want to give them chemo side effect. So
Mhm.
I will just give Pembro so that it will be as good as chemo and it's FDA approved. So that school of thought, I would say it's called physician's discretion. Like everyone has different philosophy, right? So when you categorize them, refused chemo, you can put at physician's discretion.
Okay.
So I just want to explain the 042 data to all of you so that you know the context where it's coming from. Okay.
Okay, so for these cases, I'll describe it that way.
Share screen again.
So this patient did not want the chemo, and the PDL1 was over 20. And the next patient was PDL1 40%. And then,
So is that physician's discretion too?
yeah.
Okay.
So I just got the impression that when patient did not want chemo or not a good candidate for chemotherapy for whatever reason, and when PDL1 is like reasonably high, like 40 to 50%, or if there is like at least one PDL1 level from Tempus, it kind of sounded like they just went ahead for the Pembro only.
That's what actually happens in clinic. Yes.
Yeah. So this patient had like PDL1 40% diagnosis, and then so this Pembro only was third line after cisplatin and pemetrexed and doxycycline and ramucirumab.
Third line. Wow. Oh, this must be an old PEP patient, like,
Yeah, this patient was treated in 2017.
Oh, okay.
So like patient treated back in like 2016 or 17, like sometimes there was like not even like a proper document for the PDL1 at the so and then this patient was PDL1 40 to 50%, and then given 50% in PDL1 IHC expression he was started on Pembro.
So I think that's, I don't want to call that low. It has 50%.
So
Maybe
do you want us to just
I think it's better to call them more high group. Let's call it 50%.
okay. And then the next patient was PDL1 was 25%, but performance status was kind of poor and had just moderate fatigue. So
I guess poor performance status is yeah, that's as a rationale. Yeah. Well, so that's not a good candidate for chemotherapy. So that's just to put that as a rationale. Refusing chemo, not good candidate for chemo, and then at physician's discretion, those are all good causes. Yeah.
okay. And then this patient was Yeah, so for this patient, I think the annotation was so this patient was stage 3, I think. So this patient was on tumor board for the confusing like staging thing. And then the tumor board recommendation was it looks like just stage 3A, and then
Oh.
got the radiation therapy.
Followed by Pembro?
Yeah.
For how long?
Started on October
Oh.
2018, and then progressed after 2 months.
Oh. Interesting. What was the PD-L1 level?
10%.
Well, I would say unresectable stage 3.
Yeah.
It progressed through Pembro. Yeah, I remember this case. I think we gave it like systemic therapy, knowing it's unresectable, and SBRT may not be a cure for this
Yeah.
patient. So
Yeah.
I think it's fine to leave it. And then this is more like at physician's discretion.
Okay.
Patient's probably not a good candidate for chemo.
Mhm.
But this is more like, let's use it more in synergy with radiation and all that. It's more physician's philosophy. So yeah,
Gotcha.
at physician's discretion. So yeah.
And then this one was recurrent non-resectable advanced disease. And then cardiac history, and then poor performance status.
Yeah, not good candidate. Yeah.
Yep. So these are okay. And then there are some questionable cases there. But these Nivo and metformin, there were 3 patients who were Nivo and metformin trial.
Okay.
So I think that makes sense.
So
And
that's Nivo metformin. So disregard metformin because I
Yeah.
know it's a trial, but it's you can, and for the analysis purpose, you can just regard them as Nivo patient. But it's
Mhm.
okay to leave it like that. But the reason why Nivo was selected was patient needed second, third line immunotherapy, right?
Yeah.
Oh, and then are these immunotherapy naive patients or?
I can't exactly remember, but I think there was like 1 patient who were immunotherapy naive, but adding metformin to increase the sensitivity to Nivo
Yeah.
as part of the trial.
So let's say there's a patient who got Nivo, progressed. I put the patient on Nivo and metformin trial, right? Then in that case, well, I personally believe metformin helps. That's why I wrote the trial. However, for the sake of this analysis, the metformin is not important. Like the timing of when metformin started is not important. It's like whenever Nivo was started,
Yeah.
till it stopped working, whether the metformin is on or not, doesn't matter. So that Nivo trumps any metformin related treatments.
Okay.
So if Nivo wasn't given before, but you're just starting the
Mhm.
Nivo metformin, it's fine. Just count that as Nivo and but I guess Nivo metformin in so this was before the era of Keynote 4.2.
Okay.
Not because that data only pertains to frontline. But Nivo approval was 1% or higher.
Okay.
So let's make sure that these 3 are not PD-L1 negative. Can we at least look at that?
One was 25%, two were negative.
Oh, I see. So then I would say these in three cases, let's just say all of three, the reason for immuno only in this setting was clinical trial. Let's just call it reason is clinical trial.
Gotcha.
Yeah, that's clean. Yeah, I like it.
Yeah. So
What-
some of, I think one of them for sure was started right up, like with the Nivo and metformin, like with the trial, like as a trial enrollment. Yeah.
The sad thing is we still haven't published this.
Oh,
Liam, do you know where this is? Metformin paper? Is that with Tan? Dean? Can you look into it, Liam?
Yes, it's with Dean.
Yeah. Can you follow up with him? I know he was coming and then he's not coming to our lab meeting, so
Yeah, I've been messaging him like every other week, but I'll follow up with him again.
yeah, my recollection is that it was almost done, and I think
Yes,
it's
so
forever to get it published.
just out of curiosity, like, was the result like positive?
No, there was no resist. There was
Okay.
no mixed response. There was no resist, but there seems to be, I believe, PFS benefit. Well, Liam, let's keep this in mind. The Nivo metformin paper and the Nivo veliparib paper. Maybe a good project for someone who comes to our lab, MD, whether it's Eunhee or whoever's coming. Let's have those people actually finish up and submit it. I mean, it's too much to just wait this long. Okay. Anyway, thank you, Iser, for looking up all these details. I think this is
No problem.
really good. I mean, like, I
Yeah.
feel like you are also learning like what's actually being done in clinic. Like,
I actually am. Yeah.
what is the reason they selected this and all that, and
Yeah.
kind of learning the history, like, learning the history of the immunotherapy approval over time. Yeah, good. Okay.
So for these two patients so there were these three patients who had atezolizumab, and then I think this patient was incorrectly assigned to this group because it was obviously atezo, cisplatin, and docetaxel combination treatment.
Oh, okay.
So I think this has to be corrected. And then these two,
Wait. Cisplatin seems to be given as neoadjuvant. Do you know if it was neoadjuvant, right?
so it was initially stage 2B, and then resection happened, and then
Well, I think resection happened between.
yeah, and then so that was neoadjuvant, and then atezo was adjuvant, looks like.
Okay. So good. If you look at one by one, you'll find these incorrectly assigned patients.
Yeah, so that's why I colored these four as blue. So because I wasn't sure if that has to be considered as a palliative purpose, because all these three are neoadjuvant durval, just for 1 year.
Yeah, so they are
Curative.
adjuvant. They are
Yeah,
or
yeah.
consolidation. Yeah, so they don't belong to this category.
Yeah, right. So we should not consider this as palliative purpose treatment that is included in the survival analysis.
Correct.
Okay. Yeah, so I think I'll have to remove these four from the survival analysis. All these three are the same thing.
I'm glad you looked each patient. Okay. Well, now I'm a little worried because you only looked at low group. What if in high group you have someone similar like this?
Yeah, you're actually right. So I think I can
think
like look into that too.
Yeah, I think you have to look into that group as well because of the same reason. Like, you can only find this after reading what's actually going on, right? So,
So the last question is these two patients who received Nivo only therapy. So it was just written, it was just documented like stage 4 squamous cell, and then concurrent chemoradiation followed by Nivo only. Oh, can you hear me?
Yeah. Do you hear me?
Yeah, I do, but just your video is frozen.
Yeah. Maybe because of snow, I don't know. I see myself frozen there too. So concurrent chemoradiation happens for, as you know, unresectable stage 3.
Yep. But that was just so weird because like, it was obviously documented as stage 4.
It was followed by Nivo.
Yeah, so carbotaxol chemoradiation for 5 weeks and then Nivo.
So maybe this is a clinical trial and maybe it's saying
Oh,
Nivo for 1 year or
mhm.
something.
Yeah, I think you're right. Oh, I think I can look it up and then clarify that thing.
Yeah, you can look it up and so if you find this is clinical trial and Nivo is only given for 1 year, this is not stage 4. This is on stage 3 still, and this the same thing as Durva. This is consolidation therapy and should not be part of this cohort.
Okay. And then stage 4, this one was stage 4 mucinous adenocarcinoma, and then received 2 cycles of carbo and gemcitabine as a neoadjuvant, and then it was noticed that she had another hilar lymph node metastasis, and then she was started on Nivo only.
What would that's tricky. Stage 3B carbogem neo. So never had surgery?
So after, never had surgery. So got the neoadjuvant therapy with carbo and gemcitabine.
Uh-huh.
And then before the surgery happened, they noticed a hilar lymph node metastasis.
But hilar lymph node is usually resectable and they usually just go ahead with the surgery. How do they I don't know if they found hilar lymph node mets through surgery. No, are you sure the patient didn't get surgery?
I think I'm not sure about that. I think I just checked the medical oncology note, and then it said just like after having the neoadjuvant therapy.
Patient had lymph node mets? Okay. So check the chart. I want later after this meeting. If there was any surgery done, this is neoadjuvant, and then the follow the Nivo for 1 year. Is consolidation. So it doesn't belong to this group. If it is truly stage 4 and Nivo was given forever, like not 1 year, then it's stage 4. It's okay to be in this group. But if that is the case, then yeah, the start of the Nivo would be the cycle 1 day 1 time. Yeah, but to me it looks more like neoadjuvant case.
Yeah, I can take a look at it again, but
And it progressed?
yeah, the patient progressed and then passed away after 3 months from the treatment.
From the start of Nivo?
Yeah.
That's so fast.
Yeah.
You know what? This patient, I'm really interested in knowing what actually happened to this patient.
Yeah, I'll take a look. I just kind of assumed that because like it was like initial note was like initially stage 3B and it was getting neoadjuvant, and then the later note was like um describing as stage 4B, and then they found hilar lymph node metastasis. I just kind of assumed that probably they found like some like bilateral hilar lymph node that was not there before, like during the neoadjuvant, and the treatment was switched. But I'll take a look.
Yeah. Can you? Yeah. So while we're talking, let me. I think I can just quickly share my screen. This is 0099. It's not showing up. Can you tell me what the MRN is?
Oh, for this patient. 0099
Mm-hmm.
538 64.
Okay. Let me share my screen. Do you see my screen?
Yep. And then you can go down to you can scroll down to July 2016, and like that's when the treatment was started.
July 2016.
Yep.
Oh, let's go to notes. Or should I just look at my notes?
Yeah. Yeah, the July 4th was when the Nivo was started.
So this one?
The July 4th was when Nivo was started, and then June 22nd was before it was started.
Oh, so this is when I first saw transition care. Okay, so mucinous stage 4, lobectomy.
Oh yeah, she actually got it. Oh,
Lobectomy was 2015.
okay.
Superclav node, FNA. So there was a superclav, and there's a pleural thickening.
Mm. Oh, I see.
Pleural disease made it stage 4A.
Mm.
Got Carbogem, University of Chicago, Dr. Maitland.
Mm.
Suture line. What is that? Suture line measuring small intract. Radiotherapy. Interesting. So Carbogem, 2-cycle, and they did radiation. But that was quite a while later.
Yeah.
March radiotherapy. Submeth was growing. Increased interval, increased left hilar lymph node, pleural based nodule concerning for metastasis. So this is when they started Nivo.
No, Nivo was started in yeah, but yeah.
Start with second line Nivo next week. Okay. So this is true stage 4, mucinous, usually doesn't respond very well to anything.
Mm-hmm.
This patient is superclad and pleural. Yeah, so. Wow, she's 29-year-old. Never
Yeah.
smoker. Oh my God. I feel like I remember her. And yeah, she died really young age. So it's the mucinous that's really, really
Yeah,
problem.
but that's the point I got kind of confused that like she was administered 2 cycles of neoadjuvant carbogem.
Yeah. Yeah, but before then that she had pleural mets, so she was stage 4 even before that.
Yeah,
So
then why would she get the
yeah, I'm just thinking if this was truly in, if they wanted to do surgery, they usually give cisgem, first of all.
Mhm.
And gem is usually given to squamous. So for adeno to give gem is not usual. I would do cis pemetrexed in this case, but thinking of any surgery. So this was given, I think, just to take care of metastatic disease. And it's not like they did radiation right after. And I think sometimes you do carbogem to sensitize for radiotherapy. And if
Yeah.
and then sometimes you give it together, but if her condition wasn't that good, we do chemo followed by radiation approach.
Is this neoadjuvant for like neoadjuvant for radiation therapy, like not the surgery?
So people who do not have good performance status, you do chemo followed by radiation rather than doing concurrent chemo radiation. It's not in the book. It's not in the guideline, but that's in the real world. People do that. So that's one possibility. And the other possibility is carbogem was just given as stage 4 treatment. And then as a consolidation later after 3 months, they just use radiation to consolidate. Because you do that consolidation for stage 4 patients sometimes.
Yeah.
I think that's the case, probably.
So the carbogem was not technically the neoadjuvant, and she was already stage 4.
Yeah, I think just consider that as a part of stage 4 treatment. What happened to my camera? It's not even working. What happened to everyone's camera? Is this? I don't know. Oh, but I don't.
I think it's not working, Dr. J. Oh, now I see you. Now your microphone's not working.
Okay. All right, it's working. Let's now go back to your.
So if that's in stage 4, progress after a couple cycle of cytotoxic chemo, with this devolume monotherapy, like appropriate, like does that make it appropriate like treatment option?
But it still fits our criteria, right? Stage 4 after many lines of therapy. Stage 4 therapy, it's still Nivo only. Yeah, so it's fine.
Then for this one, like how should I describe it? Like,
can you share your screen?
Sure.
Oh, so that's it's so again, this is before the Keynote 042 era.
Mhm.
So this is, you can say, standard of care for second line and beyond,
Okay.
because it's not negative, right? It's low. Was the PD?
It's negative.
Oh, it was negative. Wait. Was it approved for negative? You can put it as physician's discretion.
Okay.
Mm. Mm. Let me so I think the first approval of Nivo in lung cancer 2015, and it was approved based on everybody randomized to either Nivo versus docetaxel. Nivo approved overall survival compared to Doci, hazard ratio of 0.7, median overall survival 12 versus 9. And that was, in fact, everybody, not like based on PDL1.
Interesting.
Very interesting, right?
Yeah,
And
it was back in 2016, so that makes sense.
yeah, so that was 2016, and this was approved in 2015.
Mm.
And they did so that was the checkmate 057. So that was the Nivo approval for all comers. But Pembro did something different, that they targeted PDL1 positive in that space, and showed that the PDL1 high is the one that really receives benefit. Anyway, yeah, just knowing that history. So at that time, that was the standard of care. Like PDL1 status wasn't taken into consideration.
Gotcha.
So standard of care for second line, I think it wasn't just this one. I think there was one more that was standard of care. If Nivo was used as a second line or beyond in that 2016-17, that time period, that you can say standard of care for second line and above
Gotcha.
as a yeah, indication.
I think I'm not sure if we went through these two patients. Like these two patients went through atezolizumab only. And this patient was likely stage 4A, although we cannot rule out the synchronous lesions. And the atezolizumab was obviously started before the PDL1 came back. Then PDL1 level was negative under 1, but it was still continued.
Yeah, less than 1 means negative.
Yeah.
But it was still continued. Do you know what line? Second line, third line?
Yeah, I think I do know. So if it's first line,
Oh, it was the first line?
based on what we collected. So this one, I think I remember it, like when I was collecting the data, it was Dr. Mohindra's patient. And then the patient was new patient, and then PD-L1 level was not even back. And was started on Atezo. And then after the PD-L1 level came back negative, Atezo was continued.
Oh. So first line Atezo standard of care is 1% or greater. I think. So I would say maybe at physician's discretion, maybe is a better classification, because you don't want to normally do that in frontline.
Yeah.
Yeah. Well, now the sun is out. It's really, really clean, clear sky. I have lung cancer approval for Tecentriq. So for your I will share screen. I want to show you guys this. You see it? So Tecentriq indication is here. So if you're using an adjuvant, 1%. For the first line, you need
Acid.
high PDL
PD-L1.
Yeah.
Or
or IC immune cell 10%. So what an interesting first line. So this is Mohindra should have done this, right? If it's the standard of care. And then if you're doing combination, the PDL1 doesn't matter.
got it.
And the first indication is the last one. People who progressed on chemo. Then it was approved regardless of PDL1, just like Nivo.
Oh,
Yeah, for the second line and third line. You
I see.
you get it?
Got it.
Yeah. And there's HCC, melanoma, and alveolar soft part sarcoma.
So I'll
Uh,
triple check, like if this patient had any like chemotherapy, even like
Before,
prior
yeah.
to office visit, like from other hospital, even.
Then standard of care.
Yeah, or if there was like any other PD-L1 that was done, like other than the biopsy, I'll double check that.
Yeah.
So I think that's about it. So I can definitely go through other PD-L1 over 50 patients regimen. So if those are like consistent with the guideline, I'll just leave it like that. And if there are some questionable stuff, like I'll just like look into that. But my only concern is like as I am in Korea, like I'll be here until next Saturday. I might have to use like a VPN to make it look like I'm logging in from the United States, because obviously it has to change the password. Like if I log in from Korea, I think that's what happened last time.
Yeah.
So
Use VPN, whatever. And then the time zone's different. So maybe when you are able to look at it, I don't know. Still keep trying. It's fine. And if whatever reason you can't find it, work with Liam. Liam can look things up for you. But yeah, ask me for passwords. I'm happy to.
Yeah, so I'll try VPN or logging in different um time like maybe like after you're done for the work or things like that. And then if
Yeah,
those
but
doesn't work,
when did it
Mhm.
happen? Like when it makes you go out of the system? Did it happen to someone? It was a Juhi or who it happened to someone?
what about I don't know what you're
Like logging in from Korea.
Yeah,
When did
that was me. Like I think I logged in once from
oh,
Korea, and then that was like almost like a couple hours after you logged out, and
oh,
then the system sensed that like that's like technically not possible that I log in from Asia like a couple hours after you log out from America. So
oh,
they sent you an email to change the password.
oh, really? Okay. Okay, let's see what happens with VPN. Okay.
Yeah, and then if nothing works, I can ask like whoever in the lab, and then I can go through it like together. Or I could just work on other projects and then just finish this after I'm back. Like,
Yeah,
yeah.
Dr.
So
Teddy can
Mhm.
also use can oh, but maybe she's not able to get into Epic. Are you able to get into Epic? Dr. Teddy? Maybe she's not able to. Okay. Anyway, are you able to log into Epic from where you are, Dr. Teddy?
Uh, actually, no, I haven't tried it yet. So, well, actually, I'm not doing the EDW, so I don't
No.
need the
Uh,
access.
okay, just curious.
Oh, okay.
Okay.
But I was able to when I was trying to.
You weren't able to, right? You weren't.
I weren't. Yeah, I wasn't able to because
Yeah, I
yeah.
know. Okay. Some computers it doesn't work, but at least your laptop works, right? Ether?
yeah, it
Yeah.
works. But using the VPN, I'll have to figure out the way. But like my parents' computer does have VPN, so I might have to like log in again with that computer. I'll try like whichever way I can try. So I think that'd be best because like I'll have plenty of time like while I'm here. But if not possible, I could just like focus on the review paper I'm working with Juhi and
Yeah,
try to finish it. And then when I'm back, like we have like some off days during the ED, so I'll have some time to spend full day on the reviewing these. Um, so I think like now we change these stuffs. Like I'll have to work on that like demographic table and survival analysis like all over again. So I think like we'll have to basically change the whole tables and figures.
Yeah. I think we might have a better result because we're kind of getting rid of the ones that really skew the data, I
Yeah.
think.
But I mean like it's not a big deal actually like making the table. So I'll just make sure all the data is kind of reliable. And then my one question was last time we considered like doing the subgroup analysis based on the regimen, so just ipi-nivo only or like pembro only, and then among PD-L1 under 50 groups, like we can divide patients into very stretched good or bad, and then compare like IO only or chemo IO and things like that.
Right.
My only question was about this like regimen-based analysis. We have like 20 patients who got ipi-nivo. But unfortunately, among them, only 2 patients are very strat poor.
Only
So
2.
yeah. So I don't know if that'll give us like meaningful result. But when we did this PD-L1 under 50, these like whole patients, and then did very strat good or bad, the result was actually pretty good.
So only 2 are PDL1 low or negative?
So among PD-L1 under 50, last time you mentioned like for those patients who got ipi-nivo with PD-L1 under 50, like we can see very straightforward good is like separating the curve and all that, but only 2 patients among them are very strat poor.
I see. Okay. I think it's going to be hard. 2 versus 18 patient is so many random factors there. So I don't think it makes sense to. However, I think you can still use that as a supplemental figure.
Sure.
Still make one. I think the reviewers might want to see by regimen. And maybe not by just the low and negative, less than 50, but you've done it with all comers, right? All comers, very strict, good, poor, just the
Yeah.
niveau. You've done that, right?
Oh, like regardless of the PD-L1 level?
Right.
We did not, but we could do that.
Yeah. So that might have more sample numbers and
Mhm.
yeah. So I think people are more familiar with Pembro and Pembro containing regimen.
Mhm.
But people don't know how Nivo Ipi only treated patients behave. And you can, because I do a lot of 9LA regimen, remember?
Mhm.
So you can do Nivo Ipi only regimen. You can do Nivo Ipi containing regimen, which
Okay.
includes 9LA patients. And then look at that all comer, separate them by very strat, good or poor. And then you can look at subgroup of that analysis, which is 50% or below. So in fact, I think that subgroup analysis, like I know we've always been doing immunotherapy only, immunotherapy containing, and then looking at subgroups within PD-L1, and then look at the effect of Veristrat, right? But I think what you can do is people are now more interested in 9LA and 227, which is Nivo, Nivo Ipi only regimen or Nivo Ipi containing regimen, right? The reason is the 5-year follow-up data is superior if you compare to the Pembro regimen.
Mhm.
Okay, and that's a really big thing. And more people are now using Nivo-Ipi in lung cancer space. But no one really looked at the effect of Veristrat in Nivo-Ipi only group, right? Have you seen any paper? You're the expert in the field.
Yeah.
So I think you can do that Nivo-Ipi only, Nivo-Ipi containing group, and then in that group, what is the impact of Veristrat in terms of PFS, OS? If you have the toxicity data in terms of the toxicity, and then you can look at subgroup PD-L1 negative or a low group and say, is the Veristrat's predictability still true in that subgroup in this regimen group? Something like that. You could work on that and submit for the AACR in San Diego next year.
Sure, but would you like to like add that on our manuscript as well?
Yes.
Would that be an issue, like if in case this paper gets published like before AACR and the posters some like accepted?
So when it gets accepted and we do proofreading, we tell them the part of the paper is going to be presented at the AACR. So just embargo the paper for that poster presentation date.
Okay.
So it won't be an issue for the whole review process.
Okay.
Yeah. The more I think about it, Nivo AP regimen is a good subgroup that we could present.
Sounds good.
And the deadline is 2 weeks left.
Sorry.
The deadline is 2 weeks left. 2 weeks.
Oh, for the AACR.
Yeah.
Yeah, so I think in that case, I really have to make sure that I can collect the data from Korea.
Mm.
Um,
While you're I mean let's do it. Let's do different troubleshooting.
okay. So this is it. And then I'll be having a meeting with Judy regarding the AI Pathomics project. So
Okay.
I'll be sending the manuscript that Horin has been working on, and then the Google Doc that we have summarized like what we found in ASCO this year.
Mm-hmm.
And we're gonna start the literature search process.
Okay.
We should be able to give you an update next week.
Okay. So I found a really nice paper
Mhm.
from Lunit and from I think it's Asan. No, Samsung Group. Dr. Park Se
Un.
Park Se-un. And I know you just arrived in Korea, so I had Dr. Seyoung
Mhm.
present that paper because it used Lunit AI Pathomics. It just got published in a
Oh,
really
is that like what Dr. Chan Young posted on the Facebook? Like
yes,
with the
yeah.
biomarker, the response rate was like 70%.
Yes.
That was really cool. I thought it was really cool.
Yeah, so that has to be in your review.
Sure.
But Seyoung, can you show that paper to us?
So did they? So I think they published in JTO or like JCO. What?
Yeah.
Are you asking me to do the journal review right now, or
Right
oh,
now. I want to go through the data with you.
Also, Dr. Che, I also got a question about the AI Pathomics project.
Yeah.
So are we like narrowing the scope of like AI pathomics used as a biomarker to predict like outcome, or like AI pathomics used for like whatever purposes? Because I saw like in the University of Chicago they developed this algorithm for diagnosis itself, like not as like a predictive biomarker or anything.
Mhm.
So
So I think we for that review paper
Mhm.
there was a title. Do you have the title in that Google
I
sheet?
think I have to like dig it
Yeah.
up somewhere.
Yeah. Why don't you find it? But let's look at this study quickly first and then get back to the scope of your review. But yeah, Seyoung, I let you present it.
okay. I'll try to be fast as I can. Yeah. So this was a journal that was accepted on October 5, which is really recent. A phase 3 randomized study of atezolizumab plus bevacizumab and chemo in patients with EGFR or ALK mutated NSCLC. The summary would be the objective would be to answer the question of chemotherapy combined with anti-angiogenic agent. Bevacizumab in this case can improve clinical outcomes of EGFR ALK positive patients who already progressed on targeted agents. The knowledge generated, the result was that atezolizumab plus bevacizumab paclitaxel carboplatin, which is ABCP from now on, significantly improved PFS compared to pemetrexed and platinum combination PC, with 38 risk reduction and showed a high response rate. The relevance would be it improved ORR and longer PFS with the 4-drug combination compared to PC, but it showed higher rate of grade 3 or greater TRAEs. So for the introductions, two main therapies are introduced here. One is tyrosine kinase inhibitor, TKI, and one is bevacizumab, VEGF monoclonal antibody. So as you can see on the top right of the image, this is a monoclonal antibody that targets variety of mutations specifically. So for the NSCLC, and these pathways go down to generating tumorigenesis and progression on the down image, as you can see. So if you target this, it would be helpful. So for the NSCLC, this is the first line treatment for metastatic NSCLC in EGFR mutation or ULK. However, most patients receive acquired resistance despite the response that's shown initially. And ICIs also are showing controversial results. So for now, platinum-based chemotherapy is the standard of care for patients who has failed on EGFR TKI. And bevacizumab is also a monoclonal antibody that targets VEGF that is related to angiogenesis. So using bevacizumab or anti-angiogenic agent might enhance the activity of chemoimmunotherapy by increasing lymphocyte trafficking into tumor microenvironments. Reversing immunosuppression is their hypothesis. And this was also coming from the result of phase 3 IMpower 150 study, which showed in a subgroup that improved efficacy of anti-PD-1 antibody combined with anti-angiogenic therapy, particularly in EGFR mutated NSCLC, showed improved efficacy. However, this was a very small population and exploratory nature. So phase 3 randomized study was to be required. So this study came from that, using that result to actually see if this shows the efficacy and evaluate safety too. So going on to trial profile and material methods, I kind of try to mix it together. So this is a phase 3 multi-center open label randomized trial conducted in 16 hospitals across Republic of Korea. Eligible criteria that are major are written on the left side. It talks about stage and if the patient were to have specific mutations, what treatment the patient had to be required on, ECOG status, etc. From the screening, 254 patient was recruited. After screening, total 228 patient was to be randomized. And the mutation population was, as you can see on the left, EGFR very was very high in proportion. So ALK translocation or ALK mutation analysis was very weak in this paper. Before the randomization, the physician determined whether induction treatment would be included, and the randomization was done by 2:1 to ABCP and PC. The regimen can be found as below. This was stratified based on the mutation type and presence of brain metastasis. No crossover was permitted. After the randomization treatment, maintenance treatment was applied to both arms, and the regimen is also found on the right side. This was done until the disease has progressed or event of unacceptable toxicity occurred. And if a clinical benefit was to be anticipated, continuation of atezolizumab beyond disease progression was allowed. So from the 2:1 randomization, 3 patients were excluded from the ABCP arm because no drug was administered, and safety and efficacy evaluation was done. The primary endpoint was investigator-assessed PFS, and secondary endpoints are as follows. Imaging assessment schedules are as follows. And if a patient were to have a PFS and would have missed 2 or more scheduled assessments immediately prior to the event, the last tumor assessment prior to the missed visit was used as a center point. Other than that, exploratory biomarker, I think this is what you would want to look more. the expression value of PDL1 by SP263 and SP142 was evaluated, and patients without available outcomes, immunohistochemistry assays were done. And for the immunophenotype assessments, the LUNATScope IO powered AI was used to analyze TILs. Statistical analysis methods are as follows. So for the result, for the baseline demographics, total of 225 patients were analyzed. And as you can see on this table, a majority of the population consisted of female and never smokers. The baseline characteristics were evenly distributed between the arms. And majority of patients also had brain metastasis in both arms, which was up to 41%. And majority of patients had already previously failed EGFR TKI therapy in both arms, starting from about 92%.
So go back. So brain met is 40% is truly high. So if you have EGFR, they're more likely to have brain mets. In fact, they're more likely to have distant mets. So yeah, so that's really important.
Okay. Yes. So going over to the results, the clinical efficacy and survival analysis, this is the ORR result. Median duration of follow-up for the study population was about 26 months, and the ORR and ABCP arm was significantly higher than the PC arm. And for the MPFS, median PFS was significantly longer in the ABCP arm too. However, this was not seen on the overall survival. And this is the water plot for tumor response per RECIST. So the upper figure is ABCP arm, and the figure E, the down below, is PC arm. A significant reduction in the sum of target lesion was found. And for the subgroup analysis, this was conducted based on PFS because there was no difference per OS. And the subgroup that had brain metastasis had favor of PFS on ABCP arm significantly. And this was also seen in patient group that had L858R EGFR mutation, and patients that did not have T790M mutation.
Well, go back to PDL1. So did PDL1 have any dose response relationship here? What do you think?
It actually, are you asking me or Dr. Eve?
Both.
Yeah, I think it does because PD-L1 over 50 versus under 50.
Yeah.
They also talk about it on the backside of the, what is it? The manuscript also reports about it. So.
Okay.
Yeah, I'll go on. So this is the safety analysis. In general, the ABCP arm showed more risk on safety. Any TRAE observed was 96.7 on ABCP compared to 75.7 in PC. And also TRA leading to permanent discontinuation there was 3 patients in ABCP arm, and 2 patients that discontinued treatment permanently, and 3 treatment-related death in ABCP arm. However, PC arm did not have any at all. And for the ABCP arm, peripheral neuropathy, alopecia, and myalgia was the most common adverse effects that was found. And for the exploratory biomarker analysis, the subgroup analysis was conducted using PDL1 SP263 and SP142 expression profiles. And when it comes to PDL1 that's less than 1%, there is no difference. However, when it comes to more than 1%, significant prolongation of PFS was found. And if you look at the hazard ratio, it's 1.747. And when it gets higher to more than 10% and more than 50%, you can see that the hazard ratio also gets better, 1.41 to 1.24. And so the manuscript says PFF benefit was greater with higher cutoff of PDL1 in the APCP.
Well, go
So,
back.
yeah.
So this was, so you have to know the 22C3 is for Pembro, but 263 is for Atezo. They use different monoclonal antibody for staining. And that's 263. They like to use 10% as a cutoff. But anyway, the higher the cutoff, the better the benefit of the 4-drug regimen. Yeah.
Right.
I'm wondering if they did the same thing with overall survival. Did they?
I'm not sure. Maybe I can look it up in the supplementary and let you know later, but I'm not sure for now.
Because yeah, when you look at these papers, always look at the supplemental information. They usually do that there. And if it does show benefit, then it's a real benefit. Yeah.
But from what I know, I don't think they showed any benefit because there wasn't anything that was shown as a figure. But I'll let you know and I'll check and let you know.
Yeah.
Yes. So the result was that, oh yeah. And
Lunit.
for the TILs, using AI-powered Luniscope analysis, these were the proportions of high inflamed scores, which use 20% as a cutoff. So 1%, 19, until 50, it's 14.8, and 50, over 50, it was 37.5. And for the analysis, patients with inflamed score below 20 did not show any PFS benefit. However, patients with over 20% of inflamed score showed significant prolonged PFS compared to the PC arm.
So this is great hazard ratio 0.2. I think that was for PDL1. So PDL1
50.
was 0.24 or something. But go back to that, like, but then that was how many patients here? Only 15 and 8. So 23 patients belong to 50% or greater group, right? 23. And what about here? How many patients here? 23. So it's still very similar. But I'm surprised. This is lung cancer and 20% inflammation is only 12 and 11 out of what? What is our the mother population here?
Which?
The whole population, the arm.
Oh.
What is the 2 different arm total population? Each arm.
So
154, 74.
Right, but the ABCP was excluded by 3 patients, so it would be 151 to 74.
Okay. So still very small number.
Yeah. Right.
Okay, but anyway,
Yeah.
it's so nice separation. Okay.
Yeah, those were the results for the discussions. Because significant benefit of PFS was found in the EGFR mutation, especially for the L858R mutation, since it's associated with poor clinical outcome, this suggests that this 4-drug combination could be a reasonable treatment option. And also, patients treated with 3rd-generation EGFR TKI as first therapy showed more prominent PFS benefit. And since this is a current preferred first-line therapy and no other SOC exists after progression other than the platinum doublet, the paper also suggested this regimen, 4-drug regimen, could be further investigated in this patient population. It also made a point on the brain metastasis patients. Patients with brain metastasis had greater PFS benefits, which could mean that efficient delivery of ICI and chemotherapies to the brain was made by applying bevacizumab. And they also suggested a potential reason for the non-significant OS could be due to high incidence of subsequent post-study treatment rate in both arms. Higher grade 3 or higher TARE was most related to cytotoxic chemotherapy. And it compared the results with prior studies. So the result concorded with IMpower 150 study. And Orient 31 study in China is a study that uses PD-L1 and bevacizumab compared to other arms. And this also shows significant improvement in PFS. So it kind of concords with this result, but it still remains unclear whether the different chemotherapeutic regimens have a significant impact on clinical efficacy. And for the global trials on the bottom, this was chemo plus ICI vs chemo alone in EGFR and NSCLC progressed on EGFR PKI. However, this did not demonstrate any clinical benefit. So this paper also suggested the role of anti-angiogenic agent combined with ICI and EGFR mutated NSCLC.
Okay, so let me give you. Oh, so it's not. Oh, you have a few more slides. Okay, go ahead.
Oh yeah, and yeah. So it also made more points on combining EGF inhibition with atezolizumab regarding EGFR mutated NSCLC responding poorly to ICI, regarding low TMB PD-L1 expression and decreased TILs. And for the exploratory biomarker analysis, since magnitude of PFS benefit increased with PD-L1 high expression, they made another point on this. This was also observed in CheckMate and Keynote clinical trials too. And favorable response of patients with high inflamed tumors also indicates the essential role of bevacizumab. And it goes on with limitations of ITT, the sensor dates, monoethnicity, different in trend in patients' prior treatment and current SOC. And yeah.
Okay.
So yeah, this is the end.
Yeah, go back to your discussion. I want to just give you some points. Oh yeah, go back. Well, show me your last slide since you made it. Make sure. So what's the conclusion? What's the take-home message from?
Oh, so the conclusion is that atezolizumab and bevacizumab to chemotherapy showed a significant improvement in PFS and ORR in patients with activating EGFR mutation or ALK translocation who had already failed prior TKI improvement. Though this data, since this wasn't confirmed with OS benefit, there should be a long-term follow-up suggested. However, this regimen still offers a superior clinical benefit to pemetrexed and platinum doublets as a standard of care and should be considered as a feasible option for the subsequent treatment after EGFR and ALK TKI resistance.
Okay, so let me give some. Yeah, great job summarizing it. I think you did a great job. So anytime you design a trial, the question is it going to change my practice? Is this paper going to change practice tomorrow in my clinic? Dr. Isil, what's your answer?
I think the ABCP regimen you've been already using it. But these post hoc analysis with the Lunit inflame score can give you like more confidence, like how this patient would like respond to that regimen.
Yeah. So yeah, obviously there's many things like, oh, maybe not a good candidate for Taxol. You know, the other thing is the comparison used pemetrexed, but then the experimental arm didn't use pemetrexed. But so what I do in clinic is not Taxol, but I use pemetrexed and then continue that pemetrexed, bevacizumab, atezo. And that's what I do in clinic. So because I believe pemetrexed is better than Taxol, and you can do maintenance with pemetrexed. So that's my clinic. For EGFR patients, I've been doing it even before I had this result. So I'm gonna continue to do it. Now there is no overall survival benefit, probably because immunotherapy is used somewhere, down the road, and maybe it's affecting the overall survival. Or probably Avastin or ramucirumab, the VEGFR drug, is used down the road with docetaxel or things like that. So it will prolong survival in that way. The bevacizumab or ramucirumab are both VEGFR targeted therapies that will affect overall survival at some point. So it might not matter when you receive those drugs. So to me, OS benefit lacking is not a big deal. So and then I would based on the subgroup, more likely consider this regimen for brain mets, exon 21 mutation, PD-L1 high group, right? So that's how it's going to impact clinical care today, and I think it will go to NCCN guidelines because clinical practice changing, and it just came from one doctor from Asan Medical Center. Oh, let's do an investigator-initiated study. Let's just get these was it 16 hospitals across
Yes.
across
What I believe so.
Korea, and they enrolled about
Yes.
how many patients? 228 patients. And that's exactly what I want to do with the last trial. I want to use LUNIT, accrue 100 patients, have about 10 sites in our IIT. And if IIT can be enough to change practice and be part of the NCCN guidelines. So this is why we do what we do. Like, I have a question, like, should I give immunotherapy to EGFR patients? Never as a monotherapy, but if you combine it with Avastin, maybe it's beneficial. If you just give immunotherapy and chemo, it's not beneficial. That's, that was the other trial that was failed, that failed. So go back to your last discussion slide number. I want to give you some teaching points.
This one or
Go to the next one. Go to the next one. So the Orient 31 is a China study, and it used methotrexate, not Taxol here, right? And it did show PFS benefit. I think they don't have OS benefit, but this is a proof of concept. Sintilimab is like PD-1 inhibitor. So if you're not sure about one study, oh, it's small, it could be a random effect, look for another one that supports the same concept. So I think it's true, and they use pemetrexed, the more the reason to use pemetrexed instead of Taxol. So my criticism is, why do you use Taxol here, right? And then Checkmate 722 Keynote 789 is a trial that showed if you don't use anti-angiogenic EGFR, you're not going to see any benefit of immunotherapy. So this is why these type of study is so important to understand what's actually going on. So going next slide. So I like this one because it talks about biology. So EGFR tumors have higher VEGF, and VEGF plays a role in immune suppression. So if you block VEGF, then you might be able to do better. But then if you have infiltrated kills and high PD-L1, maybe these are the ones who will respond better with the immunotherapy. And the role of immunotherapy will only be intensified if you have the VEGF inhibitor on board. And maybe that's so true for EGFR tumors. However, what about non-EGFR tumors that will have high VEGF expression? There are going to be those tumors. And the one prime example is renal cell carcinoma, where Pembro is approved with axitinib. So axitinib is a VEGFR inhibitor. And if you could argue maybe if you do Lunit scope, you can kind of tease out who's going to benefit more with the same type of analysis. So I think looking at the role of TIL and VEGF expression may be something that could play a role. And we have EGFR tumors mutation as part of the Lunit project, but I think we excluded them. I mean, because of the sample number, we can't really look at it, but EGFR or VEGF is a big predictive marker for immunotherapy, as well as its interaction with TIL. So keep that in mind. And then the low tumor burden you mentioned here is interesting. That's a nice, very interesting hypothesis. And if you look at Lunit paper, TMB and their TIL score has some correlation, not strong, but some correlation. So I think that's also important. Okay, so with that being said, I think it's a great paper. Isa, you need to add that into your Pathomics review paper. But more and more studies are incorporating this TIL markers, and that is our IIT with Regeneron, right? Remember we met at ASCO and we talked about their combo trial. So we're gonna do the exactly same thing. We're gonna look at by different PD-L1, TIL, different TIL score. We might. So I'm a little bit discouraged that 20% was just like 10 patients out of 140, 70, and that's too low, right? We thought it could be higher than that. But I don't know why it was so low in this population. Maybe because it's EGFR tumor, right? If it's non-EGFR tumor, the TIL high group may be higher. And I think, let's say 10% or 15%, it's still amazing to see because we think tumor, cold tumor. We think so. But even that 10-15% can be salvaged by giving immunotherapy and and bevacizumab I think it's still a big deal. So anyway, I really applaud, congratulate the Samsung Medical Center investigators who made this type of trial possible and was able to work with Lunit to produce this wonderful paper. And I think it was presented at ASMO Madrid, Spain, and was on embargo and just published in JCO. So it's a really good work that is presented by our Korean investigators. So I feel like this is sort of like the good role model. A clinical question and no pharmaceutical companies interested. So you propose it as an investigator-initiated trial. You get your own funding, do the study, and explore the science with Lunit team. So I feel like this is a really good paper. And definitely deserves good review in your paper, Iser. So can you show and good job, Shaeyoung, and you can unshare your screen, and Iser, can you
Yes.
share your screen for the Google Docs title of your review?
Sure.
Horyun started. By the way, Horyun is coming next week to our lab,
Oh, awesome.
so he can continue to help you finish this.
Sounds good.
Okay, role of AI and digital pathology in the era of immuno-oncology, current status and future perspective. So in the era of immuno-oncology, so my focus for this paper is in the introduction. You can focus on, yes, it's used to used for diagnosis purpose as just an introduction. We're not analyzing it. Okay, for the diagnosis. But for immune biomarkers, let's say they use AI to say predict PD-L1 expression. That's immune oncology right there. So predicting immune biomarkers. I don't want them to predict EGFR like mutation. That's targeted therapy. So if you're predicting immune biomarker, let's say a radio the digital pathomics predicting TMB, okay, that's, to me, that's immune oncology. TMB is a marker of immune oncology at this point because it's an indication for Pembro, right? So only TMB, PD-L1, or TIL, right? TIL is an immune biomarker itself. So that, and then any AI-based biomarker predicting response to immunotherapy, right? TLS or whatever that is, predicting response to immunotherapy is the main review category. Is that clear?
Yeah, that's clear. So mm, so this is what we have found after that ASCO. So these are all like biomarker related papers. So
Yeah.
I think that makes sense.
Yes. Yeah, in Alkin, remember we went to their booth and we found something, but you can't find it.
Mhm. So all these are about the biomarkers for the immunotherapy.
So two things. One, you can just predict known biomarker like PD-L1 and TMB, right? To me, that's really not important because, well, you can just sequence it, right? You can just do IHC. Why do you want to predict that, right? To me, it's only relevant if like insufficient sample. I can't run PCR. I can't run NGS. Hemothorax, pneumothorax. I can't do more biopsy. I have to make decision, but I only have H&E slide. It's going to be helpful in that space. So in your review, write that utility. I don't think it's limited benefit. You will going to have NGS anyway for treatment decision making, but in case it's not technically feasible, clinically feasible because of these challenging clinical scenario circumstances, then it may be useful in those limited circumstances. And then describe can pathomics predict PD-1, PD-L1, TMB. But those paper has to kind of end there, not predict survival, okay? And then the use rest of the papers that predict survival to more like predicting, outcome of immunotherapy. And that's so the immuno-oncology is like you have to use immunotherapy. If it's just predicting, let's say stage 1 to 3 patients and they don't use immunotherapy, like TCGA data is an example. And if they're just predicting prognosis of lung cancer in general, that's not immuno-oncology. But it doesn't mean you exclude every paper like that. But if they are, oh yes, I predict PD-L1. That yes, I used TIL score and that predicted prognosis of lung cancer in TCGA data. It's okay to present that because it's like some sort of AI used PathOmics to find some immune signature and that has some prognostic implication. So you can still introduce those papers in the biomarker section. But the real big thing is, can it predict response to immunotherapy? So
Got it.
those are two different things, prognostic and predictive. So
it's biomarker related and then not limited to lung cancer, is that correct?
correct. It's still very new field, right? We haven't found many articles yet.
Yeah, but I found like something like they tried something at University of Chicago for head and neck cancer.
Uh-huh.
Yeah, I'll add that up too. So I'll just start working on it with Jui, and then once Horyun comes, like he can join us.
Yes, and then if the papers are really interesting, maybe prepare, just couple slides to show us what it showed when you present next week. And maybe Chui can do that too. Yeah, so let's do that. But I think they're using all different technologies that we cannot do any meta-analysis. We can't pull the data. It has to be just a review paper.
Yep.
Okay, any questions about this one?
No, I think that's about it.
Okay, so Liam, what is the status with Biodesics? Because we thought we were gonna get the raw data and we're nowhere.
Yeah, so we're updating the MTA basically.
Mhm.
Yeah, so we just have to wait for that to be finished.
So whose court is the ball in?
So right now I was able to put our legal department and Biodesics legal department in touch.
Yeah.
So they are sorting it out. There was an email just this morning where they were just kind of getting on the same page about which documents were missing and needed to be updated. So hopefully we'll hear from their progress soon.
Okay, so once that MTA is updated, we can receive
The raw data, yes.
and once it's there, then either we will work together doing raw data analysis. I was really hoping that we get something out by AACR deadline, but I don't think it's gonna happen. I think it's just taking too long to get it. So I think the Biodesics, once the MTA is done, the raw data for toxicity analysis will come out, but then there's another one that Dr. Tegu requested, right? Patient between 2000, some data was missing.
2018.
2016 January to August.
Well, did Jackie provide that data or not yet?
We recasted it multiple times, but she said she's only able to give us the accession number. And then that accession number is not working because it's like too old. So we couldn't like identify the patients like MRN or anything.
Oh. So she gave you accession number and that she said that's the only thing she can do?
Yeah. So the thing is, Dr. Wonbin added patients from December 2015. Yeah, December 2015 to December 2017.
Okay,
And then when I came, I got a data from August 2018 to 2020.
Hmm.
And then when I got those data, it has accession number, patient name, and DOB.
Hmm.
So at that time, I just added those. I just analyzed that data only, and then I've been publishing.
Huh?
And then when we try to compile all these data that we have for this manuscript, we were wondering if we could add like data like further. So we were trying to add one base data, and then we found out that there's like that 6 month gap that we don't have the data to make it like consecutive. So there's that 6 months, like 2018 January to 2018 July. So that patient list, Jackie just gave us the accession number. But once we get the accession number, then we should be able to find the report from the portal to identify the patient. But these accession numbers were not working. Like we couldn't find a report with those.
So Jackie knows that's a problem.
Yeah. So she said like the accession numbers that everything that she could give.
So Liam, do you know what the solution is? Did we talk about this? Was it well, does MTA have anything to do with this?
That was my assumption. So because once you have the MTA, it should cover all of the data points.
Right,
and
the patient
yeah,
name, right?
yes. So my thought was that once we have the MTA in place, we should be able to resolve all of the issues we have right now.
Okay.
Well, I think that patient name and stuff, like even before they brought up the MTA issue, she said like she cannot find like patient name or anything other than accession number.
Or she
I
can
don't know.
find patient name. Well, she can probably pull up the report.
Because like that's what we really, really wanted, because like back then we were really running out of time, and then we got this 400 patient list with only accession number. Then we had to manually search all these like patient report to find the name, which Taegu actually did. So for that patient before 2018 August, like we did the same thing, but for those patients, like Taegu could not find their report. So we could only add the patient after 2018 August.
Okay. So let's do this. Liam, why don't you just call Jackie and say this is what we want, and is MTA is going to be able to fix this problem? Can we get any patient data that where we can search up the patient? Where MTA is not going to fix this, you guys don't even have resource to find the patient report. I mean, if you know the simple, if Jackie can with that old accession number, if she or anybody in the company can pull up the old PDF file of the report, why don't you just send those the report to us in a the identified zip file or whatever that is. So just figure out a way. So number one, is it related to MTA? If it's not, anyone in the company be able to pull up the report of with the old accession number, or you guys don't even like? I mean, it doesn't make sense. I can't imagine a company where their accession number they don't know how to locate their old reports. They should be able to do that. And then can we get that old report? And I mean, if it's not related to MTA, we should just really force them. And I will tell them that, I'm not going to be able to work with a company that, is not able to pull up my, old patient data. And they without MTA, I think they were able to pull up data anytime. And we did very shit. But if it's an old data, if they can't pull, I'll just have to be, like really, pushing them to do it. If it's MTA thing, then we will have to push for MTA to be done. So why don't you call Jackie and find that out, and let's come up with a firm plan.
Okay.
Okay, let's do that today, Liam, and let me and Mr. know. Okay. Isu, is there anything that you're working on? You need help? Oh yeah,
Mm.
you need TLS. Where is that paper?
So TLS, like Dr. Alexiv just got us slides. And then he said rest of them are underway, but I
Underway,
think
right? Oh my God.
so I was actually gonna like email him, like, should we consider this as final and then just work on our manuscript to wrap up, or should we be expecting more?
So let's do this. Just CC me and I will email right after you email. So I know thank you for the 10 slides. I know you said there are a few more coming. Do you know by when you can get, because we really want to submit this, soon. If you feel like you don't, there's not going to be more slides, then let us know. We will just proceed with 10 slides. But if you can give us more slides, we would appreciate it. But if you can just let us know, approximated time when you'll be able to give us, that will help us, plan for the submission and all that. Yeah, let's send that email out and I will also push him. And if we come to a point we can't work with Dr. Alex, then we can't. I just have to just find another thoracic pathologist. I feel like this is too much. I feel like we're at that point where we wanna switch.
Yeah, I also feel that way too. Like it's like without getting that resolved, like there's not even point of like screening out this EDW thing, because like we wanted to add a lot of patients from that, but like we don't have the pathology department cooperative.
Right. Okay.
This is like very unusual response, because I've seen like the University of Chicago, the AI pathomics related project I've seen. So they're developing their own model to diagnose, and then they validate it with U Chicago cohort for like each cancer type. Like they made this like 400 slides like validation set, like easy, like in like less than a year. Like why does it have to take like a couple of years just to scan like 100 slides?
I feel like we need someone other than Alex. I just feel that now. Okay, but I think we have to say, is this final? Let us know.
Yeah.
Yeah, if not, give us a timeline, and then if he's not willing to work with us, then I think we just don't work with him anymore. I feel like I think we're at that point.
Yeah,
So
because like for this additional slide that we sent, it was like about 120 slides. And then like even the patients who got the biopsy in the recent 3 years, it was almost like 40, 50 patients. So if we could only add those patients to our data, like that'd be very meaningful. So I kind of wanted to wait until he gets back. But yeah.
yeah. Okay, and then remember, Liam, we were trying to have a telecon with the PathCore, the mirror, and then he was gonna find another pathologist who's gonna work for us for the last trial. Do you remember that?
Yes.
Do you know when that meeting is going to happen?
No, we have not heard back. We
Can
can
you
set up.
follow up on that email and set that meeting up?
Yeah.
And then we can make that maybe not a Zoom call. We can also make that an in-person meeting, so we get to know them, and then that pathologist can help us do what Dr. Iser is doing.
Yes. We were actually waiting for Lunit to get back to us because they were trying to see if they can visit our facility so that we can discuss the logistics together.
Okay. So yeah, so they reach out to Lunit. Are you going to visit us? If not, yeah, just follow up.
Yep.
Just follow up on weekly basis. Okay. So So Isra, if we get more slide, then we'll be able to, finish that project, right?
Sure.
Okay. All right. Enough of frustration here. I really feel like TLS is a really, really good high impact project, and we need to publish it in a timely manner. We just not been able to do it, but yeah.
Yeah, because like now we only decided to add these slides from the lymph node and then the primary site and then lymph node only if it's found to be qualified by the pathology. So now we only have like 69 slides in total. So Lunit, like really you wanted to add like as much as possible, like if it's anything possible.
I see. Yeah. So I will work on it. And maybe if he doesn't respond in 1 week, let's just find out another solution. I think we've had it. Like we waited too much. I think we have to. Okay. Anything else, Isir?
No, I think the most urgent thing for me is finding a way to do the trial review from Korea. That'll be, I think, very helpful to wrap up all these things.
What review?
The trial review, the opening epic from Korea.
So in the next 10 minutes, do whatever you can. I will send you password and all that.
Yeah, I'll have to look up like how to download that VPN thing on my laptop and all that. So
But if you don't use it VPN, you think it's gonna just happen again? Because I think I used, so when I was traveling Korea, I was using just my laptop
Mhm.
In Korea. And
yeah, so I think what you when you log in from the lab computer, like
Uh-huh.
on the computer it doesn't count, but with your laptop I think it counts. So after that happened, whenever you were traveling, I never logged in with my laptop. So, but like if you're only logging in with your own laptop and nobody else is logging in with the laptop, that's not going to be a problem.
But why don't we just, you can, I mean, like, just try it one time. You log in, and then log into my computer here.
And then if we get another email, then we find a way.
Yeah.
Yeah, well, I could do that.
And see what happens.
Yeah, I mean, it was not like a very scary email, but it was like there's like another login like attempt in like geographically like distant area, and then they recommended like you changing the password, and then you had to change the password for everything.
Right. Who got the email?
You got the email. At that time, I had no problem logging in. I worked fine. And then later you captured the email and you showed it to me, and I felt so bad. So yeah.
I see.
But I think like I could try VPN. It's like actually fairly easy. I use that for like online shopping from Korea to buy some products made in America. So I think I could try.
Oh, okay, yeah. Try, and then do you have my password? Oh,
So,
yeah.
So I think I'll have to see with my parents' computer, they already have the VPN right there because I downloaded it for them to online shop. But if I do that, then I'll have to log in again. But I'm trying to find out the way that I can do it with my laptop so I can work anywhere. Yeah, but I'll work on that. But actually, my laptop is dying. But I'm having a hard time charging it because of the voltage difference. So I think I'll head out right now. But the new charger will be here like by 5 hours. Like I ordered it coupon. So I'll recharge it and I'll work on it. I'll try to like sort it out in like next 12 hours.
Okay, yeah. And then so just messenger, and then I will give you the authenticator number as you need. Yeah.
Yeah. And I'll keep you updated.
Okay. I think we'll talk to you later.
Thank you so much.
Okay. Okay, so before going to Seong, Dr. Seti, did you have any updates or questions? I know.
Yeah. Well, for me, like last week, I have done the screening procedure, like all of them. So officially we're done with the screening parts. And after that, I've just started segmenting the peritumoral lesions. Like segmentation wise, like we're splitting the work. Like say Dr. Seong is doing the most primary part, like doing the tumor, segmenting tumor and the lymph nodes. And when she's done with it, I do the peritumoral lesions. And Dr. Monica is going to do the harmonization part.
Okay.
So yeah, we're standing there now.
Okay.
Yeah.
And you have no problem accessing the images through your computer?
Oh, I don't have any problem. It's just that my laptop is unstable there. I don't know, for some reason it's very unstable. So, but without other than that, yeah, I have no problem accessing that
Mhm.
to the OneDrive. So,
Okay. So what Seong, what is the timeline for this? Like when are you expecting?
Oh, yes. So that was something I wanted to talk to you about. I'll share my screen.
Dr. Chu,
Oh,
before moving on, I believe you have a lunch meeting today.
Right. Yeah. Just answer this question and I think we'll wrap up here and then come back to Seong later after lunch, okay?
okay. But we have the meeting at 1:30 for Picture House, so maybe after that.
1:30.
Or am I correct, Dr. Liam? It's today, right?
Yes, it's at 1:30.
And this is for me as well, right? Yeah.
Yeah, so Picture Health, now that the AACR deadline is just 2 weeks away, we wanted to finalize our like abstracts, titles, and how many we're gonna submit, and also kind of future planning. As of now, it seems like we're aiming for 2 abstracts with Picture Health, one for response and one for the toxicity pneumonitis. And Seong's been working really hard to give them like the final updated data set for this initial abstract.
I see. Okay, so so Seong, what is just one more question and yeah.
Maybe then it will be regarding the Picture Health. So the status is that we have 127 that was done with Dr. Yuri, and what I was doing was the 63 patients. For now, for the tumor segmentation, there's like 38 done, 2 in progress, and peritumor 17. So like basically everything done is like only 17 out of 63. But while I was talking to Dr. Liam regarding the AACR deadline, Dr. Liam kind of told me yesterday it might be better to switch the priority to fixing the MIP patients that are from 127 so that the Picture Health can receive these patients to include these, because for now Picture Health is using 73 only from these 127 patients because the MIP patients were ruled out, and also they were having some problems receiving the 37 files. So I wanted to have a little bit of guidance on this. That would be my important question before maybe going to Picture Health meeting.
Okay. So 127 was used for Dr. Yuri's collaboration.
Right.
Currently has 73, but if we work on MIP, So what's the timeline? Like how long? What kind of time commitment is figuring that out?
That's a good question. I'm not sure what picture. Well, for me, I'm trying my best to do as much as I can. I'm here because I have access to Epic and everything. So when I was checking with Dr. Yuri, Dr. Yuri wanted if any segmentation to be added, like these 63 segmentations, he would want it by next Wednesday. Then he said he might be able to do analysis for AAC or next 2004, I would say, next submission. And I don't know the timeline for Picture Health. And I don't think it'll be quite possible to do both the MIP and the 63 before I leave. So
what's
yeah.
the 127 and 63?
I'm sorry?
What is the difference between 127 and 63?
Oh, so 127 are the patients that were already segmented by Dr. Jian and Peter and others, and that was sent to Dr. Yuri. And you know how Picture Health.
63 is additional patients.
Yeah, right.
Yes,
These
they're
are the new.
additional.
And then did Dr. Yuri get 63 or not yet?
Not yet, because we're still working on it. Because this one starts from scratch, and this one is just modifying and fixing it. But it still takes a lot of time because the MIP patients has to be started from scratch again. And not only for, yeah.
So for 63, do we have all clinical annotation?
I'm checking it, but we have most of it.
Okay.
I did notice some are missing, but I think there's most of it.
Okay.
And yeah.
Okay, I will come back. Give me a couple minutes. I will come back.
A few moments later.
Okay, let's go back. I think that's an important question. Can you share that slide again?
Yes.
Okay, getting that MIP that is, I think should take less time than getting all 67 ready. So I will prioritize that first.
Okay.
And then we have full clinical annotation for 127, including the toxicity. So I think it's better for Picture Health to get 127. Let's prioritize that. And once that is done, we can still continue to work on 67. Now, the thing we need to know is when is Picture Health, let's say by what time when we give you the data, are you able to, produce outcome? And then by that deadline, we'll just do the new 67, and whatever we get until that day, we send out. That should be our plan.
Okay. Oh, so the Picture Health wouldn't always have to have like 127 matching as Dr. Yuri. We'll just have as much add-on as possible.
Yeah, whatever their deadline, just get whatever deadline that we have, and then combine with the new 67. Whatever we get until that deadline for out of 67 patients, combine that and send that to Picture Health.
Right, sounds great.
Yeah, because it's just ongoing work, and it's anyway.
Okay.
Okay, so I think we'll just wrap up here. Well, no, this is what I'm gonna do. I'm gonna come back at 1 o'clock, and from 1 to 1:30, let's just finish up everything that Seong prepared. So I will just leave it.
Dr. Che, you're on mute.
Yeah, I'll leave this open and come back at 1 o'clock. Okay. All right.
Dr. Che, did you need anyone to come with you to the lunch meeting today, or are you okay?
I'm okay. Well, are you able to come like the last time, Liam?
Yeah, I can rush over if you need.
Yeah. You can come over.
Okay.
Yeah. I will see you there then.
Alrighty.
All right.
One humongous meal later.
This one so that you can always go back and listen.
Okay.
Okay. Let's proceed with your sale.
Yep. Mm. Oh, just for the clinical data analysis, I'm still kind of confused because this morning you said we are not using any of the clinical variables that are analyzed here on the radiomics. But for instance, as we were talking with Picture Health, they are also using the PD, SDPR responses to predict, right? So is it just because this was previously just finished, or
No, what is the question again?
so this data of durable response, mixed response, and systematic and intrathoracic lesion comparison is a complete different project.
Right.
But okay. And
No, what I'm saying is we are only talking about Taegyu's project.
okay.
Taegyu's not doing radiomics.
Okay, but the analysis of Taegyu, like using like the result of mixed response and durable response, isn't that used in radiomics prediction models?
No. So
Oh, so that's different.
the radiomics prediction model. So mixed response is a very separate project. It's so there's radiomics predicting response and toxicity. So that's one project. And there's mixed response project where mixed response predicts survival and radiomics predicts mixed response. That's just a separate project of it itself.
Okay. Then this is just out of curiosity. Then why is this say this is predicting response? PD and SDPR.
So I don't know what analysis they've done. They have not talked to us what their definition of response is. But I think our definition of durable response has been recorded, and I think that's what we've given to Picture Health. Is that right, Liam?
The clinical data annotations?
Yeah.
We shared it with Picture.
Yeah, so I think they're just using our own definition.
Oh, okay. Okay. So there's no need to share the results from Dr. Taegyu's data to them anymore.
Dr. Taegyu's data?
Of the mixed response?
Yeah, or the durable response or RECIST or iRECIST.
So anything iRECIST or a regular RECIST based data, if we are updating it, we're updating for everybody, whether it's radiomics project, mixed response. It's a shared database.
Yeah, so that's what I'm confused about. If we are updating this, are we looking forward to sharing these updates to Dr. Yuri and Picture Health too, or is this just a separate project and not being shared? Because I was trying to ask this because I wasn't sure when we are planning to share the RECIST, iRECIST, or mixed response results to them if they were to use it?
So let me give you a. So if you look at my picture, there's a radiomics project R and
Yeah.
mixed response project M.
Yeah.
Anything that is resist is in the overlapping space. Okay.
Right.
So
Right.
any we're updating about resist, iResist has to be updated on all database, including Picture Health and Lunit, Yuri, everybody. Okay. If we find some error in a common database, then we update everybody.
Oh,
If-
okay. So we are updating them.
Yeah, we are updating everybody.
Oh,
Dr.
Okay.
Chaeyoung, did you talk about if we're gonna use Dr. Taegyu's data set or Chaeyoung's data set?
yeah. So that was the question I was trying to ask, but at first I guess I misunderstood, and so because the project's totally different, I was like, okay, we're not sharing it. And then now I'm understanding it as because it's like we have the shared database, we are sharing it. So my question is, are we gonna update all these results with the results that I came up with? Or are we going to keep the Dr. Taegyu's analysis, the data and the analysis done for the 127 patients?
So Dr. Che, right now, Dr. Yuri and Picture Health, they all have the original data set, which was created by Dr. Taegyu. And we have not shared Chaeyoung's version with anyone so far.
So remind me, Chaeyoung, what was updated based on our last discussion? My understanding is we found some errors
Yeah.
and we corrected them based on the definition of resist and iResist.
Yes. Are you referring to last Friday or like the very beginning of revising this data?
I don't remember.
Okay.
So anything that has to do with mixed response definition and then, the mixed responders, yes or no, that has nothing to do with radiomics, okay? We're only talking about toxicity and response prediction, and
Right.
that has to do with resist and iResist, how to accurately record them, right?
Right.
And that's a shared database that needs to be updated based on, reducing errors.
Okay. Got it. So we are not sharing mixed response, but we are sharing the RECIST and iRECIST results
Right.
and toxicity.
Picture
Okay.
Health and Dr. Yuri's, so for them to develop model, they only want durable response, yes or no. And we decided on the definition of durable response, right? And then we talked
Yeah.
about that's 2 weeks or something like that, right?
Yeah.
That will affect who's 1, who's 0, right?
Right. That's why I was confused.
But that's same for both projects. That's about accuracy. That's not about the theme. That's that pertains to everyone, and
Okay.
that will define PFS as well, right? PFS.
Right.
And then that PFS is going to be different. So that has to be, so Dr. Yuri and Picture Health need that updated version of our outcome.
Got it.
I'm glad- I just took it for granted that it.
Oh, okay. But one thing that worried me was that every project, like the SITC posters and everything, was based upon Dr. Taegyu's results. So, like, if we were to apply this new one, we don't know what results we're gonna get. So, and yeah, I was confused because the AACR thing is also coming up, and so we would want to share the new updated RECIST iRECIST result and the new segmentations to them, and that would be applied for the next analysis. Am I correct?
Yes.
Okay, got it.
Yes.
Dr. Che, for the SITC posters, obviously, it's too late to change anything on SITC. I assume we're not going to submit an updated poster or abstract for this because we're changing the data set, right? We're just going to update it for our manuscripts. Or do you think it's big enough of a change that it warrants a separate abstract submission?
Oh, I think we made poster, we made everything, right? For the yeah, so no need to change that. We just need to change the manuscript for that. Yeah.
Okay.
Yeah.
Okay, got it. So that problem is solved. And then yes, for the radiomics, what I've been up to is that, first of all, I remember showing you the system that I was coming up with. Is my screen showing or is it being shared?
See that? Yeah.
Oh, you see it? Do you see my screen?
Yes, it's sharing properly.
Oh, okay. So, I did made this system and then also made a SOP so that Dr. Silly and Dr. Monica can see, and so that it would help us make sure that all the most updated file would be on OneDrive. But this seems really, really complicated. So now I more understand why the 127 patient data was kind of messed up. So we are also trying to come up with the final check system so that we would make sure all the annotation and all the right files and all the right feature abstraction parameters are assigned for what we're looking for. And for now, among the new 67 patients, the DICOM series selection is done to 52 patients. A lot was helped by Eileen and also Will. Will has been helping with DICOM series selection on the new 67. Not only that, but also with the MIP patients too. So I'm getting updated on these DICOM series. And after that, I go on to check on EPIC and the segmentation list to do the tumor segmentations. For now, I believe 38 are done and 9 patients has been already excluded, which only had lymph nodes or either wrong DICOM files were assigned. And for after tumor segmentations, I'm sending this over to Dr. Silly. And prior to sending it to Dr. Silly, I've been trying to work both. So I've done until 17 and 9 is still in progress. So I'm looking forward to doing as much as I can. Probably I'll look forward to doing the MIP patient updating first. Will is also checking on the new 37 files that is not yet shared with Picture Health to see if there are more MIP patients to resegment on. So this is the status for the segmentation for now. And the 127 DICOM series check has been finished. And Dr. Saley has done a lot. She did 92 out of 127.
Wow.
So I'm trying to come up with the final list so that this could be shared with Picture Health, Lunit, anyone that we're sharing, anyone we have already shared the data. I'm just making it, I would have to work on it so that it'll be easier for them to look at.
Okay.
So working on that. And this was something else, but I think this was all covered with Picture Health's meeting. And for Dr.
I'm sorry. Before moving on, can you go back 2 slides, please?
This one?
Yes. For the 67, the 9 excluded patients, are they completely excluded? As in, there's no way of, you've checked the Epic and made sure there weren't any other DICOM files or scans that we can use.
Oh, for these, to be, I just excluded them for kind of like looking forward for the deadline for now.
Okay.
Or there are a lot of reasons these were excluded. For instance, I'm writing why they're excluded here. So for instance, if this patient had no visible CT lesion and this person only had extrathoracic and lymph node lesion, these patients might not be able to get back on our analysis for now. But if we were to use lymph nodes, maybe they're back on. And sometimes, for instance, this one, the accession number that was given is just totally wrong. So they did not put on the accession date for the chest CT, but instead they put on accession number for, what is it? The IR, like lung thorax something. So it seemed kind of confusing for me too, but I guess it kind of messed up. So for this patient to be resegmented, we would have to apply for another DICOM file or so. So it just varies by case. And I also found that there are patients that doesn't have a clinical data gathered yet. So these patients would like need thorough data collection to be segmented.
Okay. I think that is going to be relatively lower priority compared to other stuff, considering the deadline. But later on, when there is time, I think definitely fill in the clinical data, and also the patients that have the wrong ascension numbers, we can collect those and we can request those again.
Sure. Yeah, I've just, yeah, so I would definitely do another like secondary like screening to make sure which can we work on more and which can be like completely excluded for now.
Sure.
But yeah, for now, I think my priority would be segmentation, and then I'll make sure this is left and organized in the future.
Thank you. Just wanted to touch on that. You can go on. Thank you.
And yeah, so, and for these lesions, I've been really trying to make sure the lesion that I'm segmenting is correct. So as you can see here, I would annotate any lesion that I feel like that needs to be double checked. And if it is double checked, I would write the another MD or doctor's name on it. So if it were to be looked over by Dr. Liam or Dr. Yuri, I'm trying to write it down. And this Monday, I believe, I went to Dr. Yuri and checked with these lesions, and Dr. Jian also has been really waiting on for the pneumonitis harmonization and non-harmonization results, which is still not done. So Dr. Yuri wanted me to come over on Wednesday to actually run this together and see what the results show. But I guess on Wednesday, I can bring my RECIST and iRECIST data too to see if Dr. Yuri can run that and see if other results are there too. So this is planned for Wednesday. And since he knows that I'm leaving on Saturday, we are trying to meet up on Friday again to check on any other lesions that needs to be double checked or other analysis that needs to be done. And for Dr. Yuri, he said if any segmentation can be done by next Wednesday, he could do analysis he thinks it's a good, like the last timing for him to run the analysis and do the draft for ACR to ACR. But since we also have Picture Health in our hands, I'm just thinking out loud. But like segmenting the files are the same for both, so I'll just try my best to do as much as I can until Friday and share it to Picture Health and Dr. Yuri and see how it goes or so.
So, Liam, I think we have a problem, right? I think we have to differentiate this poster with picture health, right? So we might have to use different language, like different model, like picture health's model versus Dr. Yuri's model we're using. We have to, because it seems like similar patient group, same patient group with similar same outcome.
Right.
We didn't have this issue because this is the first time we're co-publishing with Picture Health. What would be the wise way to sort of make a differentiating points?
Well, for me, what I'm understanding is that the segmentations that Picture Health would be having is totally different. So that results in different feature extractions. So and their prediction algorithm is different. So the clinical outcomes are same and the data, the patients are the same, but the segmentations and the predictions are different. So I guess that's the most different thing.
Yeah. I have to so think about what language would be best to, we have to use a different language.
Right. But are we gathering them together in one paper or abstract? It's just separate, right? Or is this being thought upon?
I think it's separate. Like Dr. Che, the idea is to write one manuscript with Dr. Yuri's algorithm and a separate one with Picture Health, right?
Right. But I'm just for the AACR, they would think that, having two different paper with the same data set. Yeah. So I think we have to using the auto segmentation model.
Right.
And then you could say, I don't know. So think about the algorithm's different. I know Picture Health, I think uses vessel tortuosity or something. It is a blood vessel tortuosity based model, or whatever that is unique about their model. We need to emphasize that in the title so that we're not doing two different similar things.
Right.
Think about that.
Yeah.
And maybe remember to not submit under the same category. One more for radiology, one more for biomarker, whatever that is. You shouldn't have the same reviewers look at it. Maybe remember. Okay.
Yeah, so that's been going on. I think this question that I had is resolved after the meeting with Picture Health and prayer talks. So yeah, I think that's it.
Okay. Anything else? No?
I think I'm good for now.
I know we're not shooting for AACR, but once we have, I think Lunit, there is no need to send anything interim. Lunit, wait for you finish all the segmentation and all that. So I know this is your last week, Seong, but I feel like, so I know you have to take exam. So what is your plan for future commitment? Like, the default is yes, I have to study. I will not be in contact with the team. I think I did my part. The policy is default. We understand. You have a separate life exam to prepare. So that's default, but you could be involved as much as you want, but with that comes responsibility and everything else. So I want to know what you want to do, going forward, starting next week.
Right. Thank you for asking. And for me right now, I think I wanna take on as much take on as much like kind of role that I've been having for until now, until AACR submission date. So I am planning to come into any lab meeting or the picture health meeting possible until November 16th. And honestly, for now, I've been just really, really caught up with doing all these segmentations and lab work. So I haven't quite thought through about how much commitment or how much research work I want to pick up more or drop more regarding the JLab. But I think this would be, if it's okay, if I go back and since I will be coming here for until AACR deadline, I think before then I would have more answer. Is that okay?
When you say deadline,
It's November 16th.
November 16th, okay. So you're planning to come to lab meeting and
Yeah.
any related Zoom meetings to get this submitted by November 16th.
Yeah.
Okay, so I think that's a decent commitment. And after that, you'll let us know with whatever you want to do.
Yeah, sure.
So about the authorship, like we have a few papers coming out, and I know many people have already done many segmentation before
Right.
and all that. So Liam, looking at all these, many people who've been involved in the project, we have to figure out assign authorship for these papers. So maybe we'll talk about that after this meeting. But I think the rule of thumb is you have to write up the whole abstract and kind of take it all the way through and make the poster. Whether you go or not is a second issue. If you can't go, you just then make your poster and send it to people who are actually going to the meeting. Things like that. Okay, Taeyong, if you have if any of these abstracts gets accepted, are you thinking of going to present, or if that's too much? It's in San Diego in April.
Oh wow. What would be like the deadline to say yes to that?
There's no deadline. I just want to hear your interest.
Right. So since there is a thought of me like maybe coming back, I wouldn't say like completely no for now, but like I can't say completely yes for now if that's okay. Yeah.
It's okay. Positively consider it fine. Okay. I think that's it for today. And yeah, we'll see you at the next lab meeting for the lab meeting purpose.
Yes, thank you, Dr. J.